Muchas gracias. Buenos días a todos los que están ahorita conectados para esta sesión de nuestro segundo simposio de enfermedades por priones. Y como ya habrán escuchado ustedes el año pasado, los que están aquí por segunda ocasión reunidos y los que no, este simposio se realiza bajo un esfuerzo muy grande de todos los integrantes de la Fundación de Enfermedades Neurodegenerativas México, FENMEX, cuyo presidente, el arquitecto Francisco Mota Velasco, está aquí con nosotros también y en un momento más hablará sobre algunas cuestiones de la fundación y del simposio. Pero el objetivo principal es pues generar conciencia, ¿no? Generar conciencia de las enfermedades por priones, que si bien son enfermedades que se consideran raras desde una perspectiva epidemiológica, pues son enfermedades que cuando se presentan en alguien o en la familia de alguien dejan un impacto muy grande, ¿no? Ninguna enfermedad es rara como tal cuando nos afecta a nosotros o se encuentra en la familia o en algún familiar, y sobre todo cuando ésta tiene un carácter hereditario, como ocurre en algunas de las enfermedades por priones, ¿no? Y la intención es, pues, entender qué es el prión, qué son estas enfermedades, cuál es la manera de diagnosticarlas y cuáles son los esfuerzos que recientemente ha habido para, para generar un nuevo tratamiento. El Día Internacional de las Enfermedades por Priones es en este mes, en el mes de noviembre, y en función de ello, muchas fundaciones realizan pláticas, charlas, actividades que tienen que ver precisamente para generar esta conciencia de las enfermedades y de nuevo poder entender un poquito más este agente causal que genera un poco de inquietud incluso en la comunidad de biólogos, de médicos. Algunos de ellos todavía son un poco renuentes a la existencia de una proteína que puede reproducirse por sí sola, pero cuya presencia se puede identificar claramente mediante varias pruebas estadísticas, mediante varios hallazgos clínicos, ¿no? Y sobre todo cuando tenemos al paciente enfrente y con nosotros. Estaremos reunidos una serie de expertos internacionales en estas enfermedades, desde expertos del sistema de vigilancia de enfermedades por priones en Estados Unidos, en Europa, líderes en la generación de nuevas pruebas diagnósticas, líderes en la generación de nuevos tratamientos. Estará con nosotros una farmacéutica que estará relacionada con el desarrollo ya para aterrizar en el mercado de una nueva propuesta farmacológica, así como otros responsables de fundaciones en todo el mundo. Este es el enfoque en general de esta sesión de hoy. Y esta manera de generar un pequeño índice para iniciar con nuestras actividades de nuestro segundo simposio de enfermedades por priones. Y sin más, le dejaría la voz al arquitecto Francisco Mota, quien ha sido un gran promotor de que la fundación esté trabajando en nuestro país, de que esté en pos de las enfermedades neurodegenerativas, y el esfuerzo de esta fundación, de su creación, que ha sido, la verdad, diríamos, hercúleo. Creo que le pusieron las 7 pruebas que le dieron a Hércules, ¿no? Para que pudiera ser aprobado por los dioses. Lo bueno es que el arquitecto las pasó sin ningún problema, después de un poquito de tiempo y de esfuerzo, sin duda, pero ya está la fundación como tal activa y pues organizando estas actividades, ¿no? De nuevo, sin más, le dejo la voz al arquitecto Francisco Mota para que nos hable un poquito de la fundación y de las intenciones de esta reunión.
Víctor, muchísimas gracias. Te agradezco mucho la presentación. Estimados y distinguidos invitados y ponentes, tengan todos ustedes un muy buen día. Es un honor para mí darles la bienvenida a este segundo simposio virtual sobre enfermedades por priones. Hace casi un año fue nuestro primer simposio y fue el comienzo de una nueva etapa en nuestra fundación, en la comunidad médica de enfermedades neurodegenerativas y en sus pacientes y familiares. Hemos obtenido logros o avances importantes en este año. Se logró obtener la autorización por parte del SAT para recibir donativos deducibles de impuestos. Se obtuvo el CLUNI, que es algo que tenemos que tener todas las asociaciones civiles. Su actualización que tuvimos que hacer en marzo pasado. El DIF de León nos certificó como empresa socialmente responsable y se atendieron, que es la parte más importante, varias familias de pacientes como son 2 en Monterrey, uno de Chihuahua, uno de Guadalajara, 2 en la Ciudad de México y además de una familia con 15 integrantes atendidas en Cuernavaca y la atención en línea de un caso de Argentina y un caso de Colombia. En estos momentos, la transición y crecimiento de nuestra fundación es crucial reflexionar sobre los nuevos retos y desafíos que enfrentamos. La naturaleza compleja y progresiva de las enfermedades neurodegenerativas exige que FENMEX se mantenga a la vanguardia de las investigaciones científicas a nivel mundial. El apoyo clínico y el compromiso comunitario. Por ello, me gustaría compartir con ustedes algunos de los principales desafíos y áreas de oportunidades que se han identificado. Tenemos la planificación estratégica de FENMEX y los objetivos estratégicos son establecer el primer laboratorio de alta seguridad para el manejo de priones en México, fomentar la investigación, el desarrollo y su difusión en las enfermedades neurodegenerativas, asegurar la sostenibilidad y financiera y el crecimiento de FENMEX, incrementar la visibilidad y el impacto de FENMEX en la comunidad. Y por quinto, es el desarrollo de programas para una mejor atención de pacientes, familiares y cuidadores. Ahora bien, este es un buen día para escuchar a grandes ponentes y a disfrutar y difundir los esfuerzos para generar un mejor escenario para las personas con las enfermedades por priones. Quiero agradecer a todos los que han trabajado arduamente para hacer posible este segundo simposio, desde los organizadores hasta los voluntarios. Su dedicación y compromiso han sido fundamentales para el éxito de este día. Hago un gran reconocimiento por su extraordinaria labor a la red de asociaciones y fundaciones empresariales de la Fundación Grupo México, que nos ha apoyado fuertemente. En esta perspectiva, la Fundación FENMEX organiza su segundo simposio por priones, en el cual participan ponentes nacionales e internacionales involucrados en el diagnóstico y tratamiento de las enfermedades por priones, además de la investigación de pues, potenciales tratamientos, que esa es la muy importante. Aunado a ello, presentamos las actividades de la Fundación FENMEX y de la Red Internacional de Enfermedades por Priones, el CGD International Support Alliance. En este simposio unimos esfuerzos con el Centro Universitario de Altos, quienes desarrollan proyectos enfocados al conocimiento de la fisiopatología de las enfermedades por priones y cuyo investigador principal es nuestro querido amigo, el doctor Víctor Javier Sánchez González, que es nuestro fundador y director médico de FENMEX, y quien es a su vez colaborador por casi 15 años con la Red Internacional de Fundaciones para las Enfermedades por Priones, el CGD International Support Alliance, y a su vez con varios centros de referencia y vigilancia epidemiológica por enfermedades por priones a nivel mundial, principalmente el de Estados Unidos y Escocia. Este segundo simposio de enfermedades por priones se realizará de manera virtual, pero estamos trabajando para que el próximo año puedan venir a México la mayoría de los ponentes y se pueden hacer mesas de diálogos, donde con ello dar una mayor interactividad con las familias y pacientes, además de toda la comunidad médica que nos acompañará en estos días. Este evento representa la oportunidad de unirnos y celebrar juntos, de fortalecer lazos y de fomentar la colaboración. Espero que sigan muchos eventos en los que podamos reunirnos y seguir trabajando por un futuro mejor para toda nuestra comunidad. Dentro de nuestros objetivos para este segundo simposio, tendremos el de difundir los conceptos básicos de los priones y de las enfermedades por priones, el de presentar y discutir avances en el diagnóstico y tratamiento de las enfermedades por priones, exponer a nuestra fundación FENMEX en el marco asistencial de las enfermedades por priones, presentar la red internacional de fundaciones para las enfermedades por priones, como son CGT International Support Alliance y su colaboración con FENMEX. Con esto, les deseo nuevamente a todos un muy buen día y que aprendamos juntos un poco más de estas enfermedades por priones que nos presentan estos extraordinarios amigos y ponentes, que son los mejores especialistas a nivel mundial sobre el tema. Gracias por acompañarnos. En este momento declaro formalmente inaugurado nuestro segundo simposio virtual. Enseguida, esperemos aquí, como la marca nuestro programa, estará el doctor Víctor Javier Sánchez presentando los antecedentes históricos de las enfermedades por priones. Muchísimas gracias. Bienvenidos.
Muchas gracias y es un honor también ver que ya se conectó el profesor Richard Knight. Thanks, Richard, for making this such an early connection. I will first start as giving a brief talk about the history of prions y eventualmente yo presentaré al profesor Richard Knight, quien es precisamente el encargado del sistema de vigilancia de enfermedades por priones de Edimburgo, el Reino Unido, que es una sede del Centro de Control de Enfermedades de Estados Unidos. Pero agradecemos que esté también aquí con nosotros en la inauguración, como lo teníamos contemplado. Em, bueno, si gustas, Francisco, empiezo a hablar de una vez. Ok.
Adelante, adelante, Richard. Bienvenido. Welcome, Richard.
Thanks, Richard. I will first give a brief presentation on the history of prions, if that's okay. I guess it's gonna take me like 20 minutes, more or
Okay.
less, and then we're gonna move on with your presentation. I will obviously introduce you, but I guess I will start now because we have the clock running. So yes, I will start sharing my presentation, and then we'll move on with you. No sé si ya se puede ver la presentación. Ustedes pueden verlo ahí. Sí,
Sí, se ve muy bien.
se ve bien. Ok, muy bien. Bueno, muchas gracias. Sí, también por acá, perdone. Bueno, vamos a empezar. Sin duda, para ir avanzando sobre este tema de los precios, tenemos que ir viendo cómo es que se fueron descubriendo, cómo se fueron estudiando este tipo de enfermedades, y es la intención de esta primera charla, ¿no? Después el profesor, perdón, Richard Knight nos hablará un poco más a detalle de lo que es el prión, cómo es la fisiopatología, es decir, cómo es que genera una enfermedad, y también breve introducción a las enfermedades. Bueno, generalmente cuando uno empieza a hablar de las enfermedades por priones, a veces llevamos un rato hablando y todavía no aterriza en la mente de las personas que no están en esta área de qué les estamos plati- platicando. Las enfermedades por priones llegaron a estar un poquito más impregnadas en la mente del público por una epidemia que ocurrió ya hace algunos años, hace casi 40 años, que se conoció como la epidemia de las vacas locas. Estas proteínas pues generan estas enfermedades, pero vamos a ir viendo un poquito más cómo es que empezaron a presentarse estas enfermedades y hacer el conocimiento de los investigadores y el personal de la salud. En general, esas proteínas, y no me meteré mucho a ello porque es el tema de la siguiente plática, pues generan un proceso de neurodegeneración que a grandes rasgos implica una muerte del tejido neuronal que lleva a un deterioro en las funciones mentales. Estas enfermedades tienen una aparición característicamente identificada por una demencia, un problema motor y algunos hallazgos más en el electroencefalograma o en la resonancia, que también nos mencionará un poquito más adelante. Pero de nuevo es una afectación del sistema nervioso central y es una de las enfermedades más raras, hablando de enfermedades neurodegenerativas. La principal es la enfermedad de Alzheimer y hay muchas más enfermedades neurodegenerativas, ¿verdad? Antes de encontrar como en una prevalencia importante las enfermedades por priones. Pero aún así, estas enfermedades pues afectan a los humanos y hay varias presentaciones, ¿verdad? Estas que están marcadas aquí son por mucho las más comunes, las enfermedades por Creutzfeldt-Jakob, y ahí, de ahí empezaremos a movernos un poco, ¿no? Em, esquema lo tomo de uno de mis maestros, ¿no? El doctor Paul Brown, que sin duda ha estado muy metido en el desarrollo de este tipo de estudios y que a su vez es una práctica que a él también le gusta dar, la historia de las enfermedades por priones. Y podemos empezar hablando de que esta enfermedad tiene un registro inicial desde hace pues, más de 3 siglos, ¿no? De 1720, por registros comerciales que se tienen en el Reino Unido precisamente, se describe en algunos de estos casos la presencia de una enfermedad en las ovejas que tendían a estar postradas, a tener movimientos anormales y por la descripción que se tiene en estos registros, se asume que es una de las principales descripciones de una enfermedad por priones. Esas enfermedades afectan no nada más a los humanos, como aquí estamos viendo, ¿no? Una de las especies en donde también puede afectar es a las ovejas, a los venados, al ganado vacuno, ¿verdad? Por la encefalopatía espongiforme bovina o enfermedad de las vacas locas y algunas otras especies en general, ¿no? Bueno, la primera descripción se realizó en 1720 y estas flechas nos marcan la transmisibilidad que se ha podido encontrar entre algunas de estas especies. Si bien en 1986, comentamos que hace casi 40 años, se describe la enfermedad de las vacas locas o la encefalopatía espongiforme bovina, que es un nombre técnico, y se encontró que puede transmitirse a otras especies, animales. Se encontró lo que se conoce como romper la barrera de las especies, que fue la transmisibilidad del ganado a los humanos, lo cual generó una alerta epidemiológica importante que ya estaremos viendo en su momento, ¿no? La primera descripción de estas enfermedades en los humanos como tal, sin embargo, no se rastrea a 1994. La tenemos en 1920 por un artículo publicado precisamente por los doctores Creutzfeldt y Jacob, que hablaré un poquito más adelante, y es por esta descripción por lo que se conoce principalmente esas enfermedades. Generalmente hablamos en esta área del CJD, por Creutzfeldt-Jakob disease, como una forma así muy general de hablar de las enfermedades por priones, ¿no? Bueno, vamos avanzando un poquito con ellas. Ya comentamos que en 1720 se empieza a describir esta variante en las ovejas, que se conoce como scrapie. Scrapie significaba temblorina y es actualmente como se le sigue conociendo a la variante de enfermedades por priones en las ovejas. Em, una oveja afectada pues tiende a estar postrada, se rasca mucho, tiende a tallarse, de tal forma que empieza a perder pues su lana, ¿no? Además de que tiene sus movimientos anormales. En 1750 se hicieron descripciones de lo que sería precisamente el scrapie de más ovejas en Alemania, y esos son los datos más antiguos que podemos tener. Otro registro lo tenemos en 1998 por el investigador Benoit et al, que al hacer el estudio de los cerebros de estas ovejas al microscopio reporta un dato muy importante que es la vacuolización, que se ven agujeros en el tejido cerebral y es lo que se conoce también como un patrón espongiforme, que es prácticamente lo que vemos aquí. Esa es una imagen de un cerebro en donde los priones han tenido pues su efecto, ¿no? Estos agujeros que vemos aquí, estos huecos, son las vacuolizaciones y es el patrón que se conoce como espongiforme. En 1936, prácticamente 2 siglos después de que se empieza a identificar clínicamente al scrapie, se reporta la transmisibilidad, es decir, una abeja afectada puede infectar a otra, ¿verdad? Y esto puede generar esta afectación en un grupo más amplio de ovejas, ¿no? Pasando a 1920, tenemos la descripción de la enfermedad por los doctores Gans Gerhard Kreuzfeld y Alfons Maria Jacob. Ya lo comentamos, de ahí toma el nombre de Creutzfeldt-Jakob o Creutzfeldt-Jakob, esta enfermedad. Esta descripción se hace en 1920 con este artículo que, que podemos ver aquí. Sí, aquí está la descripción por el doctor Hans Gerhard Creutzfeldt y nos habla sobre una forma muy particular de enfermedad del sistema nervioso central. Esta descripción, voy a cambiarle aquí, nos menciona que las personas tenían un problema de memoria, cambios conductuales, había una mala coordinación y trastornos visuales, que son de los datos característicos que podemos encontrar en las personas con enfermedades por, por priones. 16 años después, en 1936, estos tres doctores, Joseph Gersmann, Ernest Streusler, Ilya Mark Schenker, describen otra enfermedad que ahora adjudicamos también con enfermedad por priones, que se conoce como el GSS, por la connotación de los investigadores que la describieron, ¿no? Se conoce como la enfermedad de Gersmann, Streusler, Schenker, y es una enfermedad que siguiendo esta descripción en lo que ellos también hablan sobre una enfermedad particular del sistema nervioso central, pero que es hereditaria o familiar, nos hablan de que es un patrón clínico un poco distinto al que ya comentamos anteriormente, en el cual hay problemas del movimiento, ataxia, signos piramidales, o por ejemplo la rigidez y la demencia. Esta enfermedad se caracteriza sobre todo por ser una enfermedad de afectación a nivel cerebeloso. Ya nos hablarán también un poquito más adelante de eso, ¿no? En 1950 y en los 60 se hace la descripción de la enfermedad Kuru, que significa también pues, una temblorina, aunque también se conoce como, la nombraban como Nagi, y es una enfermedad que se describió en habitantes de una tribu de Nueva Guinea, lo estamos viendo hasta Australia, tenemos a Nueva Guinea al norte, en un acercamiento vemos aquí a Nueva Guinea, y en este acercamiento vemos precisamente la región, por decirlo así, del Puru. En este cuadro ampliado podemos ver algunas de las tribus donde se encontraron a personas afectadas por esta enfermedad. En 52-53 fue descrita el padecimiento por los antropólogos Bert y Catherine Bent. Y en el 53 el John MacArthur realiza un reporte de la enfermedad. Esto llama la atención de investigadores en Estados Unidos, sobre todo el doctor Daniel Carlton Gagdusek, quien, aliado con el doctor Vincent Sigas, empieza a hacer una descripción un poquito más minuciosa de la enfermedad y le asignan también una causa hereditaria. Esto por el hecho de que en varias personas, varios integrantes de una familia, se encontraba esta enfermedad. Haciendo investigaciones o reportes más minuciosos, empieza realizados otra vez por el doctor Gagdusek y Sigason, también el doctor Paul Brown estuvo colaborando. Se empiezan a hacer observaciones de la tribu, los habitantes de esta tribu. Hay que tomar en cuenta de que estas personas practicaban un canibalismo ritual. Cuando falleció un integrante de la tribu, se comían a esta persona en señal de respeto, pero el cerebro era asignado de manera todavía con mayor honor a los familiares cercanos de la persona fallecida, ¿no? Esta fue una observación que tuvieron muy en mente Gagdusek y colaboradores, de tal forma que buscando lo que ellos suponían que era una enfermedad viral, pueden aislar los cerebros. Primero tuvieron que estar en mucho contacto con algunos habitantes de la tribu, convencerlos de que no realizaran esta práctica de canibalismo, que permitieran hacer una autopsia, y pudieron eventualmente tener los cerebros, llevarlos a Estados Unidos y empezar a encontrar pues los hallazgos que ahora consideramos como una encefalopatía espongiforme. Cuando empiezan a reportar estos hallazgos, otro investigador, William Hadlock, en el 59, observa estas imágenes y él hace la relación con el Scrapie. Dijimos que más o menos también por esas fechas se empiezan a reportar los hallazgos patológicos del Scrapie, y él comenta, esto es algo muy parecido a lo que yo ya vi con estas ovejas, ¿no? Se empieza a hacer así una correlación entre estas enfermedades, el Scrapie y el Kuru, y en, en el 66, Dusek y Alpers, al hacer estos estudios en los cerebros obtenidos de las personas con curros que toman una muestra, hacen una homogenización, una especie de licuado, por decirlo así, que les permite transmitirlo o inocularlo en primates, encuentran que los primates que fueron inoculados con el extracto del tejido cerebral de las personas que habían fallecido con Kuru eventualmente se enfermaban, de lo cual se reporta la transmisibilidad del Kuru en los primates, y Gakdusek acuña el término de virus lentos. Por este hallazgo, el doctor Gakdusek obtiene un premio Nobel en medicina porque reporta una primera o la primera evidencia de una demencia transmisible. Eventualmente se observará que, ahí está el dato, que no era básicamente un virus. Por muchos estudios que realizó el doctor Stanley Prusiner junto con una gran cantidad de colaboradores en su laboratorio en la Universidad de California en San Francisco, el doctor Prusiner empieza a buscar virus, ¿no? Siguiendo esta teoría del doctor Dusek, no encuentra ADNs o ARNs virales, no encuentra partículas virales. Realizó una gran cantidad de estudios para buscar primero virus, después estos microorganismos, nunca se encontró nada relacionado con, con ningún otro microorganismo, y siempre contaba la proteína que ahora conocemos como el prión. Él encuentra y aísla este agente infeccioso derivado del scrapie y lo nombra prión. Una forma, un acrónimo de infectious protein, proteína infecciosa, toma algunas palabras, las junta en un término pues muy breve y digamos pegajoso, diría yo que muy efectivo para que quedara en la mente pues, de todo el mundo, investigadores, el público en general. Acuña el término de prion, publica en una revista en Science en 1982 estos hallazgos, y 15 años después se le asigna un premio Nobel por haber descubierto un nuevo agente infeccioso, que es el prion, que es una proteína autorreplicante y transmisible, y que genera enfermedades en los humanos y obviamente también en otras especies, ¿no? Esta proteína tiene esa estructura en general, no abundaré mucho en ello porque es el tema de la plática del profesor Richard Knight, pero básicamente es una proteína que cambia su conformación y le genera la capacidad de ser infecciosa y producir neurodegeneración. En 1986 se identifica una epidemia de la encefalopatía espongiforme bovina, que son siglas en inglés, Bovine Spongiform Encephalopathy, en la que ya hemos comentado, es la epidemia que también se conoció como la de las vacas locas. Y em, en esta gráfica se observan cómo fueron aumentando los casos, ¿verdad?, de la encefalopatía espongiforme junto con los casos en los humanos, ¿no? Aquí, por ejemplo, estuvo el primer caso verificado de encefalopatía espongiforme obviamente en el ganado, en el 86. Posteriormente, un par de años después, se introduce un veto al consumo de esta carne, un veto a productos derivados del ganado como tal, em, y se introduce otro veto en el resto de Europa, ¿no? Y se ve cómo años después de que se introduce el veto, los casos en los humanos empiezan a decaer. Esas observaciones fueron realizadas por el profesor Bob Will en el Centro de Vigilancia de Enfermedades por Pruebas, que se estableció derivado de esta epidemia en el Reino Unido, en la Universidad de Edimburgo. Y pues se sigue estos casos, se realiza esta descripción y podemos encontrar se encontró, perdón, una relación entre el consumo de la carne contaminada junto con la aparición de los nuevos casos que se consideran ahora como la nueva variante o la variante de Creutzfeldt-Jakob. En 1986 también se describe el insomnio familiar fatal en un artículo que publican, y aquí está una imagen del artículo que se publica en el New England en el 86, en el mes de octubre, de insomnio familiar fatal con disautonomías, con una degeneración en el núcleo talámico. El primer autor es el doctor Elio Lugaresi, un neuropatólogo en Italia, que habiendo estudiado una familia en Italia, cuyo principal núcleo se encuentra en Venecia, hace una descripción de una familia que no puede dormir, ¿no? Que tiene insomnio, pero que a su vez tenía problemas de disautonomía. Se comunica con el doctor Pierluigi Gambetti, que también se encuentra como autor, aquí lo vemos, ¿no? De autor de este trabajo, junto con otros colaboradores, obviamente, y el doctor Pierluigi Gambetti empieza a hacer las observaciones de los cerebros y reportan una alteración en un núcleo muy específico en el tálamo. Y Pierluigi se comunica con el doctor Stanley Prusiner porque para los ojos del doctor Gambetti había una esponjiosis similar a lo que se había reportado en cerebros de Creutzfeldt Jakob, de Scrapie, y el doctor Prusiner pues le manda los anticuerpos, que es una forma de analizar o de identificar las proteínas, realiza las tinciones y eventualmente encuentra que sí hay priones en el tálamo, que es una estructura en el cerebro que favorece mucho la conectividad entre varias áreas del cerebro, pero también de la periferia, fuera del sistema nervioso central hacia el cerebro y viceversa. Bueno, encuentra priones ahí y se publica el artículo como una nueva tipo de enfermedad asignada a los priones. Lo comentábamos, en el 86 se empieza a hacer esta descripción del ganado y derivado de varios estudios posteriores que realiza el profesor Robert Will, en el 96 publica un artículo de una nueva variante de la enfermedad de Creutzfeldt-Jakob en el Reino Unido, el profesor Robert Will, que fue precisamente el encargado por muchos años del sistema de vigilancia de enfermedades por priones en Edimburgo, en el Reino Unido y en Europa, y quien a su vez coordinó por muchos años a los demás centros de vigilancia de enfermedades por priones, esto hasta antes de su retiro. Em, y junto con el doctor Ironside, que fue el neuropatólogo, ¿verdad? Y algunos otros colaboradores, pues publica ese trabajo en el cual se encuentra la relación entre las personas que habían consumido esta carne con el prión y haber desarrollado la enfermedad pues varios años después. Em, muestra la evidencia neuropatológica entre los casos de la encefalopatía espongiforme con los de la nueva variante en los humanos, unas técnicas también de identificación pues muy finas y depuradas para saber que el prión que se encontraba en una muestra de ganado en específico también se encontraba en una muestra de un cerebro de una persona que lamentablemente había fallecido por esta variante y se hace una correlación con una nueva enfermedad que se conoce como la nueva variante o variante de Creutzfeldt-Jakob. Pero también ha habido otros casos identificados por transmisibilidad en procedimientos médicos, que se conoce como el Creutzfeldt-Jakob iatrogénico, en el cual en una serie de reportes se pudo demostrar la transmisibilidad desde 1974 por casos de trasplantes de córnea, en el 77 por uso de electrodos intracerebrales, en el 85 por el uso de hormona de crecimiento. Todo esto hay que tomar en cuenta que el material neuroquirúrgico pues se esteriliza, pero el prion es muy resistente a las estrategias convencionales de esterilización, también ya nos hablará posteriormente el profesor Richard Knight, y pues operaban a las personas en la etapa preclínica. Cuando una persona empieza con la sintomatología por priones, el prion ya se encuentra ahí en etapa preclínica al menos unos 10 años antes. Se especula que incluso hasta 20 años antes. Entonces, en esta etapa preclínica donde el cuadro como tal de la demencia, el problema motor no se ha manifestado, pero ya hay priones en el sistema nervioso central del cerebro, y se interviene en ese periodo y no se esteriliza con protocolos adecuados para priones, pues puede generarse una transmisibilidad, ¿no? ¿Qué es lo que ocurrió en estos casos? La hormona del crecimiento todavía en los 80 y los injertos de dura madre se obtenían de cadáveres, ahora ya son sintéticos, las causas prácticamente ya estarían abolidas, ¿no? Pero en los 80 todavía eran fuentes de transmisibilidad y en el 2003 empezó a encontrar también la transmisibilidad por transfusiones sanguíneas, ¿no? Esos son los casos que se han podido identificar. Otra vez estamos enfocados a los humanos. Hemos hablado, y si bien introdujimos al menos dos especies que muy canónicamente también nos han permitido hablar un poco del estudio de enfermedades por priones, como fueron el scrapie en las ovejas, la encefalopatia espongiforme bovino, vacas locas en el ganado vacuno, hemos recorrido un poco cómo se ha ido avanzando en identificar algunas de esas enfermedades en las que ya comentamos, el scrapie, ¿verdad? Identificar a la vocalización, que es la pues, famosa espongiosis en los cerebros de las ovejas, en reconocer la transmisibilidad por el scrapie en el 36 pues ya hemos andado también sobre el Kuru, que se identifica a mediados del siglo pasado, el aislamiento de la proteína como tal, el prión, en 1982. Se empezaron a generar otros hallazgos que no fueron tanto de carácter clínico, identificar a una enfermedad, Creutzfeldt-Jakob, Guernsey-Schenker, el Kuru, las variantes diatrogénicas o la nueva variante y algunas variantes hereditarias. Pero también ha habido algunos hallazgos en laboratorio muy importantes que han permitido ahondar en cómo es que esta proteína se reproduce, cómo es que puede haber transmisibilidad, cómo es que afecta al sistema nervioso, pero también cómo es que puede generarse una forma de bloquearla para generar nuevos medicamentos, ¿no? Y entre esos hallazgos podemos encontrar creación de ratones transgénicos que, que portan a esta proteína. Esta proteína es una proteína que todos producimos, como tal debe haber un gen en los humanos, en otras especies, para producir una proteína normal, que también ya nos hablará el doctor, profesor Richard Knight en su momento. Y como tal se han creado modelos en animales que puedan producir esta proteína, proteína variante normal, la variante anormal, la proteína normal es la celular, el PRP celular, ¿no? Y en la cual se ha visto que es necesaria para este proceso de propagación. Ya nos hablará también un poquito más a detalle el profesor Richard Knight cómo ocurre esto. Y se han generado algunas estrategias para identificarlo, tanto en laboratorio como de manera clínica, o por ejemplo la amplificación cíclica por mal plegamiento proteica en el 2001, discriminación, algunas estrategias como discriminación espectral para identificar algunas cepas o variantes específicas de los priones. Em, Algunos otros modelos para controlar la estabilidad de la proteína y generar un modelo un poquito más certero de lo que sería la estructura de estas proteínas, ¿no? Faltará aquí mencionar el desarrollo de una estrategia diagnóstica más reciente que tendrá al menos unos 5 años, que se conoce como el RT-Quick, el RT-Quick, que es una forma que es la que se prefiere actualmente para desarrollar el diagnóstico de estas enfermedades, de la cual no dudo que el profesor Chala nos hablará un momento más, pero que también será el enfoque de la charla de la profesora Ingacer, que es una de las principales personas a nivel mundial que enfocada al abordaje del diagnóstico de las enfermedades por priones, y que faltará comentarlo aquí, ¿no? El ARTIQUIC como una de las principales estrategias diagnósticas actuales para identificar a esta proteína y eventualmente realizar un diagnóstico. Bueno, por lo pronto yo lo dejo aquí con estos hallazgos. Son más que nada hitos muy grandes, enfermedades, personas que han participado con esos hallazgos y algunas estrategias más de laboratorio que nos permiten identificarlas, procesarlas y eventualmente llegar a un diagnóstico pues, más específico. Aquí dejo esta imagen que el profesor Richard Knight sin duda identificará. Es un castillo muy famoso allá en Escocia. Esta foto la tomé yo cuando precisamente estaba en el laboratorio del profesor Richard Knight y Robert Will, y como alumno de ellos. Y obviamente, pues en los fines de semana uno se daba un tour por ahí, ¿no? Pero bueno, sin duda también es un homenaje a ellos, un agradecimiento por las enseñanzas que uno recibió en su momento y por todo el apoyo que todavía seguimos recibiendo, no nada más uno como persona, sino la fundación y de manera también muy indirecta, lo diría así, México, porque nos facilitan esta transmisión de conocimientos y están con toda la apertura para apoyarnos también eventualmente en la creación de un laboratorio para diagnóstico aquí en México. Sin más, de nuevo, dejo aquí la charla. Les agradezco mucho su asistencia y su escucha. Y si hay algunas preguntas, pues tenemos que unos cuantos minutos para, para responderlas. Bueno, aquí nos comentan, hay una pregunta que nos hicieron llegar a través de YouTube, ¿cómo lo diferenciamos de una cisticercosis? Sí, las enfermedades por priones son unas demencias rápidamente progresivas, son transmisibles sin duda, pero hay hallazgos en resonancias magnéticas que son muy característicos de las enfermedades por priones. Se puede observar una hiperintensidad en la corteza cerebral algo que no es tan característico de la cisticercosis. Ahí se encuentran lesiones enquistadas muy características en el sistema nervioso central que nos permiten identificarla como una cisticercosis. Y es una buena pregunta porque también es por consumo de carne, ¿no? El prion será por la carne de res y el cisticerco pues por carne de cerdo, ¿no? Pero los hallazgos en resonancia magnética son muy característicos en cada uno de los dos. No sé si haya otra pregunta por ahí que hayan hecho en el YouTube o que aquí mismo en el Zoom nos hayan hecho llegar, ¿no? Ok, creo entonces que son todos los comentarios por lo pronto. Y entonces, si no hay más, y para darle fluidez a la sesión, yo que podemos pasar con la charla del profesor Richard Knight, que ya se encuentra con nosotros. Em, Haré una transición al inglés. Creo que en el YouTube aparecerá una traducción simultánea. Eso sí, en inglés puede ser entendible, ¿verdad? Pero bueno, Richard, I just finished my presentation on the history of prions. I know you will talk in a very, very good way, in a very good manner about prion. You may also talk about the history of prions, but first, me, the Francisco Mota, the Mexican Foundation, thank you. We are very grateful for having you this day for another session of our symposium on prion diseases in Mexico. I will introduce you. And Professor Richard Knight received his BA degree in philosophy, politics, and economics at Oxford University. Following that, he got his medical degree and his postgraduate medical qualification and became a fellow of the Royal College of Physicians of Edinburgh in 1993. Professor Knight is presently serving as director of the United Kingdom National CJD Surveillance Unit and has a personal chair in clinical neurology at the University of Edinburgh, with an honorary consultant clinical neurology post in the Department of Clinical Neuroscience, Western General Hospital, Edinburgh, Scotland. Professor Knight spends approximately 50% of his time divided between CJD surveillance and research, and 50% devoted to clinical services and teaching. He has authored and co-authored many CAD-related papers related to sporadic, variant genetic, and iatrogenic forms of the illness. He has a long involvement with CJD lay and charity organization, currently being chair in the United Kingdom National CJD Support Network Management Committee, and an invited friend and advisor to the CJD International Support Alliance. I will do it in Spanish, Richard, and then you will start. Bueno, el profesor Richard Knight obtuvo su grado de Filosofía, Economía y Política en la Universidad de Oxford, posteriormente el grado de médico y neurólogo, y actualmente es un fellow del Royal College of Physicians de Edimburgo. Él es el director del sistema de la unidad de vigilancia epidemiológica, ha sido director de la unidad de vigilancia epidemiológica para el Creutzfeldt Jakob en el Reino Unido, y es titular de la silla de neurología clínica en la Universidad de Edimburgo, con una mención honorífica también como neurólogo consultante clínico en el Departamento de Neurociencias Clínicas en la Universidad de Edimburgo, en Escocia, y destina su tiempo, prácticamente la mitad está en la investigación, la mitad en la parte clínica, y ha sido un autor de bastantes artículos relacionados con temas básicos, de ciencia básica, perdón, en las fases por priones, pero también las variantes en el aspecto clínico, donde prácticamente ninguna variante de la enfermedad ha quedado fuera de su ojo clínico. Y a su vez es muy activo, es una persona muy activa en las fundaciones, es director médico de la Fundación de Enfermedades por Priones en Escocia, es miembro del grupo de asesores internacionales para las enfermedades por priones del aspecto científico y también con las fundaciones. Y sin más, les dejo la voz con el profesor Richard Knight. Richard, we're ready to start. And again, thank you for being today with us.
Well, thank you for inviting me. Can you hear me okay?
We can hear
And
you
and you,
very well, and we can
you
see your presentation.
okay, good. Well, thank you. Well, thank you very much for inviting me, and I will endeavour to proceed, but I'm afraid in English. So, prion disease, you've heard a little bit about this already from Victor, but it's gone under various names, as you have heard a little bit about. But prion disease is the modern name, although you still hear people talk about transmissible spongiform encephalopathy and Creutzfeldt-Jakob disease. There are diseases of animals, and you've heard a bit about those, but I'm not going to talk about them. I'm going to talk about some background biology, talk about the prion disease mechanism, and touch a little bit on diagnosis and treatment, but only very briefly, because you're going to hear more about those later from others. So if we turn to the background biology, and I'm going to apologize to those of you who already know this, but I think it's important to get some basic background to understand prion disease. So the body is made of organs like the heart, the brain, the lungs, and the organs are made of cells, and everything is made of chemical structures. Now, most cells are very small, but eggs, for example, like hen's eggs or eggs in frog spawn, are giant cells. But all cells have the same kind of structure. They have a surface membrane that holds all the contents inside. They have a central nucleus, and then surrounding this, there's a fluid called cytoplasm that contains a lot of biological machinery. If you open up the nucleus, you find it contains a lot of these like wriggly structures you can see here, and these are chromosomes. If you open up the chromosomes, you find that they contain items called genes. And genes are made of DNA, and DNA is a whole string of chemical units weaved together to form a kind of double helix structure. And DNA is a set of instructions for making proteins. And proteins have structural roles in the bodies. So collagen, for example, is responsible for the integrity of our skin and for tendons. But proteins also have functional roles, for example, hemoglobin, which carries oxygen around the blood. Well, when you look at the DNA structure, it consists of lots of units, and each of these units has a code, and each unit of code is called a codon. And the DNA codons all correspond to different amino acids. Amino acids are little chemical structures, and I've illustrated three of them here: valine, methionine, and alanine. And each code codes for one amino acid. So when you translate the code of the DNA, you get a whole string of amino acids. And this long string of amino acids is the basic protein structure. It's not entirely simple because the process is done through a messenger of RNA, and the double helix is kind of transcribed to a single helix, which then is translated to the amino acid chain, the basic protein structure. Now, the genetic code instructs the production of this basic protein chain, but once that chain is formed, it then is folded in a very complex way to make the final protein structure. And this is a very complex folding in 3 dimensions. I can only show you 2-dimensional representations, but I've shown hemoglobin down in the left-hand corner here, and you can begin to see that this simple linear chain of amino acids can be folded in quite complicated ways. The 3D shape of a protein governs its interaction with other chemical structures and therefore its functions. In a way, chemical life is a series of chemical structures interacting with each other, and the shape of proteins affects how they fit together and interact. So you can see here a structure of 3 triangles, and then another structure comes along which has a triangle, and so it can fit in quite nicely. On the other hand, a structure with a circular globular structure is not going to fit in. And the shape of these proteins, therefore, is really governing how they behave, how they interact. And this is important because this interaction, which I've illustrated in cartoon form here, could be an interaction which causes disease, or it could be an interaction which prevents disease. So leaving proteins in general, we now move on to one protein in particular, the prion protein. And the prion protein, often designated PRP, is one of our normal body proteins. And the responsible human gene for making it is called PRNP. Its functions are a bit uncertain, but just to be clear, all of us, including me now, are busy making molecules of this protein, prion protein, all the time. And it obviously has some role in our body. It's normal. So let's turn now to the disease. Well, the structural main molecular event in prion disease is a protein structural change. The normal gene produces the normal protein, which is indicated here on the left, and it then converts to an abnormal form. And I should stress it's the same chemical structure, the same line of amino acids, but a different three-dimensional shape. And what is very important in prion diseases is that once the abnormal protein is there, it enables it to interact with normal protein and change the normal protein into more abnormal protein, which then changes more normal protein into more abnormal protein. So there is a self-amplification process that is going on. And once the abnormal protein forms, there's a cascade leading to more and more of it. And this is the core prion concept. A protein that can replicate itself and produce more of itself by interacting with a normal version of itself. So this is the basic process. DNA is transcribed to produce RNA. It's translated to produce the normal prion protein, which, however, in disease converts to the abnormal form. And then the abnormal form has an amplifying conversion of the normal protein to more abnormal protein. And the normal protein is usually designated PRPC, and the abnormal protein designated PRPSC. Now, the abnormal protein has a different shape and therefore different physicochemical properties. It's much less easy to break down and it's much less soluble. So it gets deposited in tissues. And you can see here this kind of pinky brown stain, which is staining abnormal prion protein, which has been deposited in the brain of somebody with one of these diseases. Now, although this is the molecular side of things, why people develop symptoms is because neurons, brain cells, malfunction and die. And in some way, all of this protein abnormality causes toxicity to neurons, and therefore they become ill and indeed are lost, and people develop symptoms. So that's the basic process, what sets it off. And we have at the moment a vision of three causes: genetic, acquired, and idiopathic. So let's deal with genetic. And this in many ways is the simplest thing to understand. If a gene is a string of code, changes could occur in a string of code by accident. And there are two important types of change: pathogenic mutations. These are changes in the prion protein code, which gives rise to a resultant protein that's dysfunctional and causes disease. But there are also polymorphisms, if you like, dialects in the language of genes. The resultant protein is functional. It doesn't actually cause disease. So let's deal with pathogenic mutations. Well, I've put the scheme here, and I put a star against the PRNP gene to indicate that it's abnormal, and it produces a different kind of protein from normal, which I've given a star to, just because the code is different. And this different type of protein is in some way unstable and is more likely to convert into the abnormal form, which then, of course, converts more of the normal into abnormal, and eventually neurons malfunction and die. Now, the original names, and you've heard a bit about this from Victor already, we have genetic CJD, Gerstmann-Streusler-Schenker syndrome, fatal familial insomnia, and so on. But I think that we're turning more and more to a much more modern nomenclature, where we call these diseases things like E200K or P102L. What does this mean? Well, if you look at the codons, the code from the DNA and RNA, you basically number the code from left to right. And I've put here 12 units of code, although, of course, the actual length of the code might be hundreds or even thousands. Each of these gives rise to an amino acid. However, if there is an error at one point in the code, then you put the name of this mutation according to which number codon it is. And the change in the code will be a change from one amino acid to another. And each amino acid has a letter symbol. So E200K means there's a change at the 200th item of code. And the change is related to two amino acids, glutamic acid and lysine, which for reasons I've completely forgotten, are encoded by the letters E and K. Now, when you have genetic disease, it is subject to what's called autosomal dominant inheritance. So if you have two parents designated by these two blocks at the top, one of them in white has a normal gene code, and the other in this sort of brownish color, grayish color, has the abnormal gene. In autosomal dominant inheritance, every child they have has a 50-50 chance of inheriting the abnormality. Now, I've put down here that they've got 4 children and 2 of them are affected and 2 of them aren't. It's very important to understand that's not necessarily the case. Every time they have an offspring, there is a 50-50 chance. They could have 4, all of whom inherit the abnormality. 4, when none of them do, they could have 3 that do and 1 that doesn't. Every time you have a child, there's a 50-50 chance. So what about polymorphisms? Well, the resultant prion protein that comes from a polymorphism is not disease causing. But of course, it can cause trouble. So let's just take an example of a code for your holiday departure. The correct instruction is that the plane leaves at 2100 hours. A polymorphism would be your plane leaves at 9 p.m. It's exactly the same time, but put in different terms. And if you turn up at 2100 or you turn up at 9 p.m., it's exactly the same result. A pathogenic mutation would be your plane leaves at 1900, in which case you arrive late and you miss the plane. Now, polymorphisms do not directly cause problems, but they sometimes incidentally make problems more likely. And certainly, in my view, if you say that the plane leaves at 9, there is indeed a possibility that some people might think that might mean 9 a.m. rather than 9 p.m. And 2100 hours is much less likely to be associated with a mistake. Now, when I said that polymorphisms are not disease-causing, but they're quite common, and in the prion protein gene, there's one common variation at position 129 relating to 2 amino acids, methionine and valine, M and V. And you see here a pie chart showing that about half the normal UK population are MV, just over a third of us are MM, and about 11 or 12% of us are VV. It does sometimes make problems more likely, and I will come back to that a bit later. So acquired disease. Well, this in a way is also simply explained because abnormal protein from an external source enters the body of the animal or the human and causes the normal prion protein to change. It's very important to understand that in acquired disease, people are exposed to this external abnormal protein, but they develop symptoms of disease later, after a period called the incubation period. And the incubation period for many ordinary infections like the cold or COVID may be very short, measured in days. But in prion diseases, it can be very long and can be measured in years or even decades. And that makes exploration of acquired disease quite difficult. The three sorts are Kuru, variant CJD, and iatrogenic CJD. I'm not going to talk about Kuru, you've heard a bit about it from Victor. I'm not going to talk much about variant, which you've heard a bit about from Victor. But basically, prior to 1989, when food restrictions were put in place in the UK, nearly 500,000 infected cattle entered the human food chain. These are the figures for variant CJD, and you can see that despite the big exposure of the population in the UK, there were relatively few cases, only 178, although, of course, that was a tragedy for those people and families affected. We have not seen a case with onset since 2014. France, being our geographical neighbour, was then next most affected. Their last case reported in 2021. Other countries have had very few cases or none at all. And to my knowledge, Mexico has had none. Iatrogenic CJD, you've heard a bit about this as well. Duramater, derived from cadavers used in surgery. Pituitary gland hormones derived from cadavers and then used for the treatment of endocrine conditions. A handful related to neurosurgical procedures and corneal transplantation. But actually, in the history of the world, iatrogenic transmission has occurred relatively rarely, with the exception of pituitary gland hormones and duramater. Just as an illustration of the incubation periods of these diseases, synthetic human growth hormone has been used since 1985, but the incubation period for CJD is long. So even though the risk went away a long time ago, we still see a few deaths, 6 deaths in the UK between 2015 and 2023. And for human growth hormone, incubation periods of 40 or more years have been described. So idiopathic. And idiopathic is a very important form of prion disease. It seems to be, as far as we can work out, due to a spontaneous error where the prion protein sort of makes a mistake and that leads on to the disease. It's called sporadic disease because it's been found everywhere in the world where it's been looked for with no particular pattern. It's the commonest form of human prion disease, accounting for over 80% of cases. In most countries, it results in 1 to 2 deaths per million per year. But if you look at the risk in a different way and ask what is the lifetime risk of anybody developing sporadic CJD in their lifetime, it's 1 in 5,000. It seems to be a spontaneous disease. We've never found any external cause. There are variations in the clinical and pathological picture, which I think will be touched upon by other speakers later. But I just want to mention, again, the polymorphism. I've said it sometimes makes problems more likely, and whether you are MM, VV, or MV does affect your probability of getting sporadic CJD. So, for example, if you look at these columns here, the green columns are the normal population in the UK, and the red columns are sporadic CJD. And you can see straight away that if you are MM, you are more likely to get sporadic CJD than if you are MV. In addition, it affects the way that the disease presents if you do get it. So sporadic CJD in these different genotype people does look somewhat different. And just touching upon this, because sporadic CJD does vary, people have divided it up into subtypes with differences in the clinical picture, the pathology, and the diagnostic test results. And they correlate quite reasonably with either the genotype, the 129 polymorphism, MM, MV, or VV, and also whether you have one of 2 types of protein, 1 or 2. So you'll hear people talk about MM1 sporadic CJD and MV2 sporadic CJD. However, from basically from the family or ordinary clinician's point of view, these are all forms of sporadic CJD, and they just affect people somewhat differently, with differences in the clinical presentation and the test results. So I'm just going to finish on a couple of brief comments, firstly on diagnosis. And what I would like to say is that the initial symptoms of prion disease are non-specific. And so when people first present, CJD may be one of the diagnoses considered, but there are other possible diagnoses to consider, and some of these are treatable. And it may take the clinician some time to work through these, but it's obviously very important that they do. There are two definitive tests, finding abnormal prion protein in the brain, but that requires brain tissue, and it's therefore either done at autopsy, which is late, or biopsy in life, and biopsy of the brain is undertaken fairly rarely. Finding a pathogenic mutation in the gene, for example, from a blood test, in someone who's clinically ill is certainly a definitive test, but it refers to genetic disease only. There are, of course, clinical diagnostic tests, and initially it's important to realize that when the people receive blood tests or an MRI of the brain, the initial reason for this is to explore other possible diagnoses. Later, people will be doing tests to support the diagnosis of CJD if that's what they think it is. And when you hear about these diagnostic tests later, as you will, you may bear in mind that some of them may be essentially non-specific. So things like the MRI are very helpful, but the abnormalities seen on an MRI are not confined to CJD. They can occur in other diseases. But there may be essentially specific tests, and a whole series of tests related to detecting abnormal prion protein are clearly specific because they are detecting the basic problem in the disease. And finally, the possibility of treatment. And I think we are coming from a time when there's been no hope of treatment through some false dawns to real dawn of treatment. We are now poised, I think, to at least reasonably expect that we have some ideas of treatment in the near future. And I just want to give you a diagram so that if you hear about treatment later on, as I think you will, it's important to try to understand the basis for treatment, and rational treatment follows understanding of the disease process. So if you look at this, you could, of course, try to reduce the normal prion protein, the PRPC, because if you reduce it, there is no or less prion protein available to be converted into abnormal prion protein. You could try to prevent its conversion. You could try to get rid of the abnormal prion protein, and you could somehow interfere with the toxicity that's affecting neurons and try to protect and preserve them. For a whole variety of reasons, which you may hear about later on, reducing normal prion protein has been the focus of many of the recent considerations of treatment. I just want to comment on one thing. In general, with disease, the earlier the treatment, the better the outcome. But early treatment requires early diagnosis. And symptoms may well begin some time after the disease process starts. And there is an important question, and it specifically relates to sporadic CJD, the commonest form of prion disease. Is it possible to diagnose it early enough to expect treatment to work? So I'm going to finish there. My road with this disease has gone on for over 40 years. And as C.S. Lewis said, day by day, nothing changes. But when you look back, everything is different. And it certainly is. The understanding of this disease is much greater than when I first met it. The diagnosis is much better. And we finally do have real promise about treatment, even if we don't have any actually proven effective treatments. So I'll finish there and hope that that has been helpful to you. And I will try to return the screen back to where we were, if I can work out how to do that. Stop share. Thank you.
Thank you so much, Richard, for your talk. I don't know if we have questions from. Yeah, I guess we have one. Igual, Fernando, si quieres preguntarla tú, porfa.
Do we have any protective polymorphisms?
Ok.
Yeah,
Creo que ya lo está leyendo el doctor.
yeah. Can you read the question? Or I mean, can you see it? It says that we
Yes,
have
I
protection.
can. Yeah. Yeah.
Yeah. But
Um,
I will translate to Spanish very quickly. Tenemos tipos de protección y es así como es que funcionan. Hay un cambio en la estructura tridimensional de la proteína. Esa es una pregunta. Con el polimorfismo del cordón 129, ¿cuál de ellos, BB, tiene un inicio de síntomas a edades más tempranas? Got it, Richard. Go on.
Well, going back to that graph I showed you, clearly MV 129 MV is partially protective, because certainly people who are MV get sporadic CJD less often than you'd expect by chance. There may be other protective polymorphisms, and certainly in Kuru, it's been shown that there's one protective polymorphism. So yes, what that means, I'm not really sure. It may help us understand something of the disease process, but I mean, let's be clear. These have a relatively modest effect. So CJD, sporadic CJD, is commoner in people with polymorphism, but a third of the UK population are MM. And of course, most of us do not get sporadic CJD. So in some ways, it's a relatively small effect. The other question about who gets things earlier, yeah, the age of onset is related a bit to the codon 129 type. But again, it's a small effect, and there are other factors which govern age of onset. These are all relatively small effects, I think.
Thanks, Richard. Have you seen any effect with any other genes? Let's say we know that apolipoprotein for Alzheimer's has some risk, is a risk factor. I mean, when you talk about E4, the E4 polymorphism for Alzheimer's, have you seen something like that for prions?
No, except that Simon Mead has done some major studies, genome-wide assessments, and he has picked out one candidate which looks quite likely to have a role. But I think all of these probably have very small roles, actually, certainly even less than the 129 polymorphism.
Okay, thanks. I don't know if we have more questions from the audience. ¿Hay más preguntas por parte de la audiencia en YouTube o aquí por el chat? No. Well, I guess not. Let me show you, Richard. We will obviously send this by email. We have a certificate
Thank you.
of your talk. I don't know where I put it. I guess lo puedo ver aquí. Yeah, it says FENMEX presents a speaker certificate to Richard Knight for sharing his invaluable knowledge as a guest speaker during the second symposium on prion diseases, which took place on November 4th at Mexico City, Guadalajara, and León, where we are all established as a foundation. And again, Richard Knight, Professor Dr. Richard Knight, thank you so much for enlightening us this day on this sometimes bizarre aspect of the prion proteins. Thank you so much, and
Thank you.
we'll be very happy, hopefully, to have all of you in a face-to-face presentation next year. We are looking forward to that. Hopefully, we can get it for 2025. Thanks again.
Yeah, thank you.
Thank you, Richard.
And now we will move on our agenda to have, and I'm very honored again to have with us a Professor Ingaser, who she's always very open to help everybody in the prion world, especially the Mexican, right here. Thanks, Inga, again for being here with us. The Mexican Foundation and me, of course, are very grateful of having you today in our second symposium on prion diseases. I will introduce you in English and then in Spanish. Professor Ingaser has a medical degree from the August University of Göttingen and a qualification in neurology from the same university and a professorial qualification from the Department of Neurology at the University of Göttingen. She's the head of the Dementia and Prion Research Group at the Department of Neurology at the University of Göttingen. Since 2005, she's the professor of neurodegenerative diseases. Since 2006, she's the head of a specialist outpatient clinic for neurodegenerative diseases, head of neurochemistry laboratory, Department of Neurology in Göttingen, head of the Clinical Dementia Center of the Department of Neurology of the University of Göttingen, and she's also been a very strong motor force in the prion field. She's very focused on the clinical aspects of prion diseases, but also the diagnostics of prion diseases. And I guess we couldn't have anyone more talented and qualified and with a major expertise for diagnosing prion diseases as Professor Ingaser. Tenemos a la doctora Inga Ser, profesora Inga Ser. La distinción de profesora en Europa es prácticamente como un profesor emérito. Es la máxima distinción académica que se puede ofrecer en Europa y precisamente la profesora Inga Ser pues es parte de este selecto grupo de investigadores con esta distinción. Es médico con una especialidad en neurología por parte de la Universidad de Göttingen y desde el 2001 ha sido la líder del grupo de demencia, enfermedades neurodegenerativas y de priones en la Universidad de Göttingen. Es profesora de las por las enfermedades neurodegenerativas desde el 2005. Es líder de la clínica del manejo para enfermedades neurodegenerativas del 2006. Es la titular y líder del laboratorio de neuroquímica del Departamento de Neurología de la misma universidad y del Centro de Demencia para enfermedades por priones, a su vez de que es un motor muy importante en esta área y yo creo que no podríamos tener una mejor ponente. Es una experta en el área del diagnóstico de enfermedades por priones, además de que apoya también en la red internacional de las fundaciones y a toda la persona que esté en la necesidad de ahondar más en estas enfermedades. Sin más, le dejo la voz a la profesora Ingaser. I will now leave the microphone to Professor Ingaser to hear her presentation on the diagnosis of prion diseases. Thanks so much.
Yeah. Yeah, thank you very much, Victor. Can you hear me? Is everything working? Can you see my screen?
I guess it's a black screen right now.
Can you see something?
No, we only see a black screen.
Then it didn't work. So I need, oh, I'm sorry, then I need to go out.
We can see it now. We can see it now. We can see your screen.
Okay. So maybe it's just a delay. So this now?
Now it's black again.
There should be, it's black again. And here? You cannot see anything. Okay. So then I go to the other mode. So then I cannot do it in a presentation mode. I'm sorry, but then we need to
It's
deal
okay.
with this somehow.
Yeah.
Okay, but then I will do it a little bit bigger. Sorry, we need to adjust it because otherwise it doesn't make much sense.
Don't worry.
Okay, so is this okay now? You can see
Yeah, we can
my slides?
we
Okay.
can see your slides with no problem.
Okay. So yeah, thank you very much for inviting me again. And Victor, I'm so really happy that this is already the second symposium. So then for sure will be a third and the fourth. And it's a very good development, I think. So I'm really happy to be part of it and to contribute. So you asked me to talk about diagnosis of human prion diseases. And to start with this, I would like to show a little bit of history, how it developed with a diagnosis of the disease. So in the 70s of the last century, we had brain, we needed the brain investigation of the brain tissue to confirm the diagnosis, either by autopsy or by brain biopsy. Then some years later, it became clear that certain changes in the electroencephalogram, like periodic sharp and slow wave complexes, are very contribute to the clinical diagnosis of the disease, and the EEG became part of the clinical criteria. Later, we had the cerebrospinal fluid. I will talk about this later. So we had the cerebrospinal fluid, then we had the magnetic resonance imaging, and we have now also tests like, which are seeding assays, which are very specific for the human prion diseases. So let's start with the cerebrospinal fluid. And when we are doing cerebrospinal fluid in patients with rapidly progressive dementia, of course, we want to exclude a diagnosis of a treatable disorder, for example, encephalitis, or meningitis, or any other autoimmune encephalitis. So there are many reasons to do the lumbar puncture and to analyze the cerebrospinal fluid. First, for some basic tests like white blood cells, the count like proteins, like specific antibodies, and also oligoclonal antibodies, and so on. So this is the reason why initially the cerebrospinal fluid was done in Creutzfeldt-Jakob disease. What we need to say at this moment is that all these tests, so the routine cerebrospinal fluid tests, they are negative or normal in patients with prion diseases. So normally we do not see a lot of abnormalities, except for maybe a little bit of the disturbance of the blood-brain or the blood-CSF barrier. So there is a little, slight elevation of proteins in the cerebrospinal fluid. But normally the cell count and all these parameters like immunoglobulins are negative or normal. I think this is important to remember because, of course, some other tests which we are using in the cerebrospinal fluid for prion diseases, they might become positive in patients with, for example, brain inflammation. So this is what we need to exclude first. So this is the first step, to do the routine CSF analysis. And then it continues. So first we know that that the cerebrospinal fluid is normally is not very or is not abnormal. And then the search started to look for some specific or more or less specific biomarkers which can support the clinical diagnosis of CJD. And this is the work which started in a laboratory in US by Michael Harrington, and he reported that doing two-dimensional gel electrophoresis of the cerebrospinal fluid, which means to separate proteins by their charge and by their size, he reported two proteins which he called P130 and 131. And these proteins he could see in patients with Creutzfeldt-Jakob disease and herpes encephalitis, but not any other conditions. Later on, There was further work to confirm that these proteins belong to the 1433 protein family. Oh, I'm sorry, I need to.
Esperamos un momento.
Okay. I'm back. I tried to do it. So then they were confirmed as that they belong to the 1433 protein family. And for these proteins, we had antibodies, and the test was much simpler. So we could do it by using Western blots or ELISA. We know that the proteins 1433 are detectable in sporadic CJD in a vast majority of the cases, or 89%, but they are less sensitive in variant CJD and in genetic cases. So this is something which has a long history and was finally accepted as part of the clinical diagnosis. So we had the first biomarker for Creutzfeldt-Jakob disease. Of course, there was an ongoing discussion that this biomarker is not specific because 1433 proteins, the elevation of 1433 proteins is probably just reflecting cell death. And there was an ongoing discussion if these proteins are specific or not because they reflect brain damage. These are data from a multi-center European study which investigated 1433 specificity in different laboratories across Europe. And we could show that the specificity is quite high in diseases which are really differential diagnosis of Creutzfeldt-Jakob disease, something like Alzheimer's disease, dementia with Lewy bodies, other neurodegenerative diseases. And it is slightly lower in inflammatory, paraneoplastic, stroke, and epileptic fits. So these are conditions which really needs to be excluded before we trust this 1433 test. So we need to know the routine cerebrospinal fluid analysis, and we need to know some clinical information. If, for example, the lumbar puncture was done after stroke or after epileptic fit, then of course it is it increases or it decreases our specificity. There are also other proteins which are elevated in the cerebrospinal fluid in patients with different forms of brain diseases. For example, in patients with, for example, this total protein tau, which is normally used as a biomarker in Alzheimer's disease, but we know that we have extreme elevation of tau protein in patients with Creutzfeldt-Jakob disease. And this elevation is not that high, for example, for p-tau levels. And normally we know, for example, in Alzheimer's disease, we have this typical constellation, high tau, high p-tau. And in CJD, it is opposite. So we have high tau, but low p-tau levels. So this is also a potential biomarker to be used to support the clinical diagnosis. And there was an ongoing discussion about which biomarker is better to be used in a clinical context, tau or 1433. And I would like to say, at the end, both can be used, but what we should be aware, I would like to show you on the next slide, is that high tau levels, for example, above 1,250 picogram per milliliter, which is calculated as a cutoff level for CJD, these levels, these very high levels, can be detected in patients with Alzheimer's disease. But they can also be detected in acute chronic vascular events, inflammatory diseases, and some other status epilepticus, for example. So very high tau levels are also not very specific for Creutzfeldt-Jakob disease. Of course, they can be found in CJD, but there's also a number of other conditions which also can present with these high levels. So at the end, probably both biomarkers have their value and both can be used, but maybe this is not really a discussion which of both biomarkers should be used, but rather than it should be used in the right clinical context. Okay, so moving from this biomarker, I'm sorry, to magnetic resonance imaging, we have a typical pattern which we can see in patients with Creutzfeldt-Jakob disease. And for example, these are is high signal in the caudate nucleus, for example, but also sometimes in some of these cortical areas, what you can see here in the occipital areas, but also in the temporal lobe. And what we have seen is that there are different different patterns we can see in these patients, and this also corresponds somehow to the molecular disease subtype. So what you can see on this slide is these are patients with different with prime protein type 1, and they have CJD, they have they have high signal in caudate nucleus, but also in the cortical areas. There are other subtypes like MB or BB with protein type 2. They have cortical areas, but also thalamic changes. I'm sorry. And okay. So this is about the MRI. And in MRI, we have seen that there are different, of course, technical requirements for the MRI, but what we know today is that diffusion-weighted imaging are the best and the most sensitive images which we should do when we think of the diagnosis of Creutzfeldt-Jakob disease. Okay, so but this is of course not the end of the story and of the diagnosis. And what was the next step was to look for a test, a potential biomarker, which really reflects the characteristic of the abnormal prime protein in patients. And as this is already been discussed, the prime diseases are protein misfolding diseases, and there are different models how it happens. We know that this happens in not only prime diseases, but maybe also in other conditions, and we have misfolded protein which forms fibrils, and this misfolded protein originates from the native patient-derived protein, which changes the conformation. So this is the story and the idea behind it. And what I would like to show you now is a little movie which comes from the year 1958. And this is a film which was made by Lionel Penrose, and he showed very, I think very interestingly, what we currently think how the test of the for the detection of the abnormal form of the prime protein works. And I hope that I can show you now this film.
Has typically been made in a series of experiments by L and R Penrose. They began by making a kind of crystal that was formed in response to seeding or priming by a small part of its cell. Each of these models, like those that follow, is simply made out of plywood. The pieces here are all alike. Each has 4 hooks. Energy is supplied by horizontal agitation in a confined space. But as you see there, the units don't link up. A linked pair is added, and the result now is that all the pieces join together in response to the agitation.
So this is basically how we think the seeded aggregation assays we use to confirm the diagnosis of Creutzfeldt-Jakob disease work. So the development started 15, 20 years ago by Claudio Soto, and he developed the PMCA, protein misfolding cyclic amplification. And there is another technique which is more commonly used. It was developed by Byron Corgi, the real-time quaking-induced conversion. And it works like in this short movie I've shown you before. So we have our body fluid where we think there are PRPSC seeds, and we add recombinant PRP. And according to this, they react, and the recombinant PRP changes its shape. Then they make fibrils, and by high frequency shaking, we can disrupt these fibrils, and then we have new fragments for this for this conversion. So we run the test for 72 hours, and then we have enough of fibrils. Then we can visualize by fluorescence dye, and then we can see this type of curve, which shows us that in the sample which we had before in CJD, there was an abnormal PIPS seed. So this is how the test works. It is now used in a clinic to confirm the clinical diagnosis, or to confirm the diagnosis. But of course, before we introduced it into the routine, we did some validation. So we checked for the influence of temperature, and we put a sample for up to 8 days at room temperature or at +4 degree fridge on, or we took samples up to which were stored up to 9 years in -80, and there was no change in the results. So the sensitivity is the same. But what changes is when we have blood contamination in the cerebrospinal fluid. So from 1,200 erythrocytes per microliter, and the test becomes negative. So the conclusion is the test is very stable, but it is very sensitive for blood contamination. So also for that, it is very important to see what the routine cerebrospinal fluid analysis has given us as information. We did some ring trials to compare the results, which was quite good. We have good results with these laboratories, and at the end there was an agreement to amend clinical diagnostic criteria by adding positive RT-QuIC in a cerebrospinal fluid or any other tissues into the clinical criteria. So this is cerebrospinal fluid, and what I would like to show you now is a first development in peripheral tissue was the detection of prime protein. So when we look in the literature, we can see that with these high sensitive methods, we can now detect PRPSC in different human tissues. It's not only in the brain and spinal cord and cerebrospinal fluid, but it's also in the olfactory mucosa, cranial nerves, in different kind of muscles, in skin, spleen, lung, and so on. So it seems that there is also a peripheral, so it is possible to detect abnormal PRP also in other body fluids or tissues, and therefore we were looking for a non-invasive procedure to test to see if we can achieve the diagnosis by analyzing, for example, tear fluid. So why tear fluid? So it was just by chance, because a colleague of mine, Paul Lingo, who is here on the photo, he was interested in tear fluids in Parkinson's disease because it's easy to collect, it's not invasive, it can be done even by a lay person, so no medical personnel is needed, and he was just interested about the proteomics of the tear fluids in Parkinson's disease. And then the idea came to let us test if we can do tear fluid analysis for the abnormal PRP. But of course, tear fluid is not cerebrospinal fluid. It is a very low volume we can collect from a patient, so only a few microliters. And we were thinking we need for this low volume, we need a better test. So a colleague of mine, she was PhD at that time, she synthesized different substrates for the test and introduced, for example, the human E200K mutation into the recombinant protein. And another protein was made using the FFI mutation. The protein, and she also did a full-length human PRP and also hamster sheep PRP as a reference. And the outcome of this was that the FFI mutation tended to self-aggregate, so we could not use it further because it was really hard to work with it because of the tendency to self-aggregation. But the full-length PRP with human E200K mutation worked quite well. And here are the major results. So what we can see here is we can detect PRP seeding activity in tear fluids of patients with prion diseases. This is the blue curve. We can also see, this is just for comparison, that this substrate we can also detect prion seeding activity in cerebrospinal fluid. And of course, it works much better with cerebrospinal fluid. So we have this curve here, which starts already after 20 hours. And the reaction is much faster and much more aggressive. But this is clear because this is cerebrospinal fluid. And of course, we have probably much more seeds in the cerebrospinal fluid than in tear fluid. But with giving the reaction to time for testing, we can also detect it in this in tear fluids. So we had 3 tear fluid cohorts. The first cohort was our discovery cohort with CJD patients, genetic cases, 26 controls. And then we selected 2 other cohorts to confirm our observation. And we tested, of course, a number of controls with different kind of individual diseases, healthy individuals, but also other neurological and neurodegenerative diseases. And these are the results of this testing. So for in sporadic CJD patients, we could detect seeding activity in tear fluids in 75% of the cases. So 3/4 of them are positive. Same number for genetic prion diseases, and luckily no controls are positive for the with this assay. So 100% specificity and 75% sensitivity. So what we also did was that we had some tear fluids collected from healthy mutation carriers. So not only from patients with different kind of genetic diseases, but also healthy mutation carriers. And because they are coming to our hospital for, how to say, as part of a prospective observational cohort, and they are healthy. But what we can see here in the curve, you can see this is the it is the acronym HMC, so healthy mutation carrier. And here, for example, for we have here 3 healthy mutation carriers for GSS, for Gerstmann-Streusel-Scheinker syndrome, and they have a quite similar curve than as our patient with the GSS mutation. Quite similar observations for the FFI mutation, D178. These are cases and healthy mutation carriers, case and the healthy mutation carrier. And also there are also other mutations where we could also confirm this observation. Which was, at the beginning, was quite shocking because we thought this RT-QuIC assay will give us the definite diagnosis of the disease. But it seems that it becomes positive in healthy mutation carriers many, many years before they get the disease. So all of our healthy mutation carriers are still healthy. So in this table, we need to add to this 35 months, we need to add another 18 months because this data originate from that time point. So up to 4 or 5 years before disease onset, they can have a positive test. And we don't know if this is disease related or if this is just the instability of the protein which carries this mutation, and this unstable protein is much easier to be converted into the pathological form. So we don't know this for the moment. But what we need to discuss in future is that we have something like diagnostic biomarkers, like Atiquic, like 1433, like tau, maybe also the MRI. But we might also have the same biomarker can be, depending on the clinical context, can be also an at-risk prodromal biomarker, which is now what we can see from the healthy mutation carriers. So they can be positive in the test, but they are healthy. So in this context, a positive aggregation assay tells us something about the disease risk, but not about the disease. This is just a hypothesis to discuss, but we need to think of it. Yeah, and therefore, and this is my final slide, therefore what we need, of course, now are biomarkers, which also can help us in this situation, in the clinical situation, that we have a patient at risk, maybe also with positive Atiquic assay, but we need to determine the time point when the disease starts. And then this is something where we are coming back to the fluid biomarkers like 1433 tau, or in this case NFL, GFAP. There are different biomarkers which might be of interest. And I think at the end we will have a kind of a at-risk biomarker, and also maybe a biomarker which really shows us that there's start of the pathology, so that there's a disease onset. I just would like to show one example. These are data from a case report, and a patient, or at that time individual, who came to our prospective observational study for some years, and we collected plasma and measured different proteins in plasma, like GFAP, tau, NFL, YKL40, and S100B. And what we could observe later was that at the time of conversion, it was this here after time point 4, at clinical disease onset, we have a elevation of NFL in plasma, which of course then with progressive disease increased further. So this could be, or this is an example of a potential biomarker which can help us to discriminate the disease onset, specifically in this situation of healthy mutation carriers and disease onset. Yeah, this is what I wanted to share with you. I would like to thank my colleagues who have done all this work and are very enthusiastic in the field, and I hope we can continue with this work for some time. And I will be happy to answer questions. Thank you very much.
Thank you very much, Inga, for such a wonderful presentation. How earlier from clinical onset do you think Artiquick could give us a positive diagnosis from, I don't know, sporadic or any of the genetic cases that you've studied in your samples?
So I think what I think is in the context of sporadic CJD, this is probably a diagnostic biomarker. But in the context of a genetic CJD, I don't think it is the Atiquick is a diagnostic biomarker, because I think it reflects the instability of the protein. And what we see is it becomes positive, yeah, in tear fluids. We don't have CSF 5 years before disease onset, so that's what we don't know. But we from the tear fluids, I can tell that 5 years and they are still healthy. So back 5 years from now, so 5 years before, they are still they were positive for the prime protein in tear fluids, but they are still healthy. So it could be also 10 years or 15 years. So the future will tell us, but we don't know for the moment. And I think what we need to understand is that that the aggregation assay is not just a biomarker. We also need to be very careful. Is this a sporadic CGD, genetic CGD? What is the clinical context? You know?
Yeah. One of the things that sometimes we do when doing a clinical study with patients who have or positive for a mutation is that when these mutations are 100%, I mean, they have a complete penetrance, then you will expect that a carrier will eventually develop the disease. So you can follow the preclinical cases just to track how these biomarkers change or fluctuate.
That's,
So
that's what I think. This is what we need to do now, is to see if there is a change of these biomarkers, and if there are some biomarkers which become positive. I don't know, hopefully, close to the onset or even before, because of course there's an underlying pathology. But I don't believe that the RT-QuIC will help us, because it is already positive, I don't know, maybe 5 or 10 years before disease onset. So the question is it really positive because it is pathology in the brain, or is it maybe positive because it is just, you have this you have the protein mutation, and it is already, it's flipping back and forth. So you don't know this.
yeah, and maybe if you combine the RT-QuIC with the tau or the 14-3
Yes.
or the NFL, so you can have like a kit of
Yes.
biomarkers that after some study you can say, well, 5 years before clinical onset, it's RT-QuIC and tau and NFL
Yeah.
will lead you to a diagnostics of a preclinical case. And obviously, when you compare this with the mutation carriers, this will be very helpful because they will eventually develop the disease whenever the mutation is has a full penetrance.
Yes. Yeah. Exactly. This is what I think how it should go. To identify those other biomarkers which might be predictive for disease onset, and very closer to the time point of the clinical onset. And of course, there will be, for clinical trials, this will be very important, because of course you will probably not treat somebody who is healthy, and it's hard to do it. Also, because you don't know, you cannot predict the individual disease onset. We know this variability of the disease, of the age at onset. It can, even in the same family, it's variable. It can be 10 years more, it can vary by 10 or 20 years between individuals. So you don't know which factors.
Have you compared any of these imaging biomarkers or with neuropsychological assessments just to try to see when one is changing, if it correlates with biochemical marker?
So we don't have so many data for imaging biomarkers. So far, those who are in our study, they are negative. So we don't see any changes in the MRI. I know there are some publications from other groups when they did the serial MRIs in mutation carriers, that it can be, how to say, some abnormalities can be detected earlier. I think there's one publication up to 1 year before clinical onset of the disease. So this is, but I think we don't have so many data for this to that we can use it in the clinical decision making for the moment. These are more or less case reports. So we need to do more on these healthy mutation carriers.
Yes. I have one question from the audience. It says, I understand that prion diseases have non-specific symptoms. In your experience, have you noticed any particular characteristic that can differentiate prion-related dementia from other types of dementia?
I think the the most clear how to say characteristic is the how rapid this disease evolves. There are not so many diseases which have really this speed of deterioration as prion diseases, and I would say this is something which develops in weeks, not in months or years, what we know from other dementia, but the changes are really very fast. And what we also see, there's a kind of combination, I would say, if you have something like dementia plus ataxia plus or cognitive decline plus ataxia plus myoclonus, there are not so many differential diagnoses. Yeah. So therefore, I think so. This is always what I think. If somebody tells me about a patient with dementia and ataxia, this is my first differential diagnosis. Could be a prion disease.
Okay. Okay, thanks. We have a couple of other questions, but I guess those are more focusing to treatment, and I guess we can put it on the next for the next speaker who's going to focus on novel therapeutic strategies for prion diseases. So if there are no more questions, I will also show you our certificate. No. I guess I'm going to have to share my screen now.
Yeah, I need to stop it. Yeah, okay.
Thanks. Okay, so I'm sharing now, and I will go to over here. Oh, I thought I had it ready. Okay, so yeah, we will obviously email this to you, but FedEx presents a speaker certificate to Professor Dr. Inga Ser for sharing his invaluable knowledge as a guest speaker during the second Symposium of Prion Diseases, which took place on November 4th, 2024 at Mexico City, Guadalajara, and León, where the foundation is established. Thank you so much, Inga, for your presentation and for being so open to guide us into the diagnosis of prion diseases. Hope to see you in a face-to-face talk very soon. Hopefully for next year. I guess we can do that for next year. And you obviously will be invited to be with us again.
Yeah, thank you very much, and I'm sorry for coughing. So yeah,
Don't
see you.
Don't worry about
Thank
it.
you.
Have a good day. And
Inga, thank you very much.
Yeah, thank you.
Okay, so I guess we came to our to the end of our first block. Creo que llegamos al final de nuestro primer bloque. Y como tal, creo que tendríamos una pausa de 10 minutos para regresar a las 12:15 con nuestro siguiente ponente, que será la doctora Nina Overbeck, que hablará sobre nuevas estrategias de tratamiento en las enfermedades por priones. Les comentamos que hubo un pequeño cambio en el orden de los ponentes por cuestiones de cambio de horario que ocurrió en Estados Unidos. Un ponente que estaba programado para ser el siguiente, que es el doctor Michael Gershwin. Producto de este cambio de horarios, vamos a tener que recurrir un par de presentaciones después, que nos hablar sobre la clínica de las enfermedades por priones, y los demás ponentes accedieron a hablar de manera anticipada. El primero será la doctora Nina Overbeck sobre el tratamiento de las enfermedades por priones, y posteriormente será la presentación de la Red Internacional de Enfermedades por Priones por Susan Solvins y Dina Simpson. Muchas gracias a todos. Vamos a cerrar este primer bloque de transmisión en un momento y regresamos en 10 minutos. Thank you so much to everybody. We will close this block of presentations in a couple of minutes, and we will come back in another one in just 10 minutes from now. Thank you so much. Thanks, Inga. Thanks, Francisco, the president of our foundation, for having all of these speakers to be with us today. Gracias al presidente de la fundación, al arquitecto Francisco, por sus esfuerzos para tener a todos a los ponentes al día de hoy. De nuevo, gracias a Inga por la plática de hoy. Nos retiramos de este bloque y regresamos en otro en 10 minutos. We'll be back. Thank you so much. See you soon. trabajó también en Gate Bioscience. Nina nos va a hablar hoy sobre algunas de las estrategias novedosas para el tratamiento de las enfermedades por priones y sin más le dejamos el micrófono a Nina. Nina, we're ready to start. Thank you so much for joining us. Mr. Francisco and me, thank you so much for being today with us.
Thank you so much for the invitation to talk today. It's a pleasure to be here. Thank you for the invitation. My name is Nina Oberbeck. As Victor mentioned, I am the Senior Director of Translational Sciences at Gate Bioscience, and we are a small molecule drug discovery company based in San Francisco in the USA, and we are working to develop a pill to treat prion disease, which is the topic of my talk today. So a small outline of the next 25 minutes. Firstly, I will give you an overview of Gate Bioscience's approach in prion disease. And as I said, we are developing a pill with a novel mechanism to eliminate prion protein. I'm also going to spend a bit of time talking about small molecule drug discovery. Why does drug development take so long? And what are the different steps that we need to take on this journey to developing a new therapy in prion disease? And I want to do a bit of education on what that process looks like. And then lastly, if I have time, I want to look ahead to the clinic. And I want to talk a little bit about what we can do as patients, as families, as advocates, as everyone working in the space. What can we do to help improve the chances of success of bringing a pill to treat prion diseases to patients? So firstly, a short introduction. As you all may know, there are many major historical names for human prion disease, and there are many different etiologies or causes of prion disease. But at a fundamental level, prion disease is caused by the misfolding and aggregation of host-encoded normal prion protein. So we all produce a lot of this protein called PRP, or prion protein, in our brains. We all express a lot of this protein. It's actually not got a very well understood function, unfortunately. Scientists don't really understand much about what this protein does in normal homeostasis. But what we do know is that occasionally this normal prion protein can misfold, and it can take on a pathogenic form or an aggregated form, a misfolded form. And when it does so, this misfolded form can, when it touches normal prion protein, it then induces normal prion protein to misfold again. And this essentially initiates a chain reaction where this misfolded pathogenic form of prion spreads throughout the brain. And this can lead to cell death and neurotoxicity. And this is what leads to the devastating symptoms that we know for prion disease. And this misfolding can happen for several reasons. The most common etiology of prion disease is sporadic. So this misfolding happens spontaneously for reasons we don't really understand. But in 15% of cases, mutations in PRP can actually increase the propensity of prion protein to misfold. And so this is the genetic form of the disease. Now, as you can probably imagine, therapeutically, we want to get rid of this pathogenic process. We want to eliminate this misfolded form of PRP. However, traditional drugs, such as small molecules or antibodies, that would bind directly to misfolded PRP. This really isn't the best way of tackling this disease. When the protein itself is pathogenic, something binding directly to it, a small molecule or an antibody, will really struggle to eliminate it. And this has been a real problem in therapeutic development for prion disease up till now. It's a reason why we've understood the molecular mechanisms of this disease, but it's been very difficult to develop a therapeutic up to now. So what's the solution to this problem? The solution, in our opinion, is to eliminate prion at the source. So if we prevent PRP, normal PRP from being made, then there's nothing for the misfolded PRP to spread and aggregate throughout the brain. And this will stop the disease in its tracks. And so that's really the new aim of therapeutic development, I think, so far in the clinic. So how do we do this? How do we eliminate normal PRP? There are several therapeutic approaches that aim to do this. You may have heard of a company called Sangamo Therapeutics. They are developing a gene therapy. And this gene therapy is eliminating PRP at the DNA level. So if you delete the DNA encoding PRP, then you no longer produce normal PRP and you no longer produce misfolded PRP. Another company is called Ionis Pharmaceuticals, and they are developing a different technology called antisense oligonucleotide technology, and that is eliminating PRP at the RNA level. And what I'm going to tell you about today is our approach, Gate Bio's approach. And this is a new small molecule to selectively prevent PRP from being secreted or being made. And ultimately, what all of these three different approaches do is eliminate normal PRP and thus prevent misfolded PRP from spreading throughout the brain. And what you get as a result is no cell death and no neurotoxicity. And this is what we hope to be a promising new approach in treating this disease. So the first question we ask is, will eliminating prion protein work? And the evidence so far in the scientific literature suggests absolutely yes. And there are many pieces of evidence that suggest this. The first is that if you remove normal PRP in mice and you attempt to induce prion protein misfolding by inoculating the brains of those mice with infectious prion, those mice never ever develop the disease. So without PRP, patients should never develop prion disease. That's the hypothesis. I mentioned also Ionis Pharmaceuticals that are developing therapeutic oligonucleotides to treat the disease, and they have published wonderful work showing that lowering PRP levels in mice can extend the lifespan of prion-infected mice. And what's key is that the earlier that you can lower PRP levels, the better. But in all of these studies, we see that if you lower PRP even after symptom onset, you still see a therapeutic benefit. The second question that we always ask is, well, as I told you, we all express a lot of normal PRP in our brains. How do we know that eliminating it will be safe, right? I told you that we don't really understand the normal function of PRP. But again, there is a lot of evidence suggesting that the answer is yes. Fortunately, we can make genetic knockouts, or we can basically eliminate PRP in a bunch of different species: mice, rats, sheep, goats, cows. And in general, this is very well tolerated. So as I said here, goats and cattle with no PRP are healthy. And actually, there's also humans that have been described that lack one copy of PRP, and they are also completely healthy. The only thing that we need to keep an eye on is that mice that lack PRP, so that are knockout for PRP, do have a little peripheral neuropathy as they age. But this is something that we can definitely monitor for in clinical trials. And so, in my opinion, and our opinion at Gate Bio, is that really this eliminating or lowering PRP levels is the best therapeutic hypothesis we currently have to treat this disease. So switching gears now, I want to tell you about molecular gates. And this is the small molecules that we are developing here at Gate Bio. And as I said, these are small molecules that can selectively eliminate extracellular proteins. Now, I'm going to try to give a very simplified overview of our technology. and then show a little bit of data and hopefully convince you that this is an exciting approach in prion disease field. So cells produce many, many proteins. Those proteins can live either inside the cell or outside the cell. Proteins that are destined for outside of the cell are called secreted proteins. And prion or PRP, normal PRP, is a secreted protein. So its destination, it's made inside the cell, but its destination is outside the cell. And prion is one of 4,200 proteins that are secreted. And in order for these proteins to be secreted, they have to go through a common entryway or a common gate, let's say, and it's called Sec61 or the secretory translocon. And it's really the common entryway into the endoplasmic reticulum through which all of these secreted proteins, including prion, need to travel through in order to be made and end up in their final destination. As I said, they must all pass through this common entryway. So our small molecules at gate, they bind directly to this common entryway, and they can selectively prevent prion protein from entering the secretory pathway. And once our small molecules block PRP from entering the secretory pathway, PRP is mislocalized into the cytoplasm, so inside the cell, and the cell recognizes PRP as being in the wrong place. And the cell has dedicated machinery that allows it to be degraded and thus eliminated. So essentially, PRP is shuttled to the garbage can of the cell where it can be degraded and eliminated. And what you get in the end is a lowering of PRP levels or an elimination of PRP, which, as I mentioned at the start, is the therapeutic goal of what we're trying to do at GATE and what other people are trying to do at Sangamo and at Ionis. So this is a bit of a schematic of what's happening here. So as I mentioned, we've managed to make several small molecules that eliminate PRP in the brain, and we have tested these molecules in mice. And this is some of the exciting data that I want to share today. So we call this in vivo target engagement. So this is the ability of our small molecules to lower prion protein levels in the brain. And the experiment that I'm describing here is we take mice and we feed them some of this compound, MG813, as you can see here, for 14 days, once a day. So we essentially mimic, you can see a little schematic of this mouse taking a pill. So they essentially take one dose a day of this compound, MG813. And we also, of course, administer a vehicle control because we want to properly control this experiment. And at the end of 14 days, we take the brains of these animals and we take the colon of these animals and we look at the percent of PRP reduction that we see compared with vehicle. So the first thing to note is that these animals tolerate the drug very, very well. We're looking here at percent body weight change over time. After 14 days of 50 mg per kg of MG813, you see no weight loss at all in the mice compared with vehicle. This drug is very, very well tolerated. And now looking here on the right, after 14 days of treatment with this drug, we see 50% decrease in brain PRP. And in the colon, we see almost a complete elimination of prion protein after only 14 days. And it's pretty amazing that we are able to achieve this just with an oral pill. And what I didn't mention is before, when I was talking about Sangamo and about Ionis's therapy, for example, that is administered via an intrathecal injection, so an injection into the spine of patients. And our approach, as I said here, is through an oral pill, which should be much more convenient and easier for patients to take. So of course, this isn't our final drug. There are still many things that we need to improve. And we hope to also see even more profound prion lowering in the brains of mice and then ultimately in humans. So we're kind of on this journey to improving the pharmaceutical properties of this molecule and making sure that we can make a safe and effective drug for people. So in conclusion, I hopefully have taught you a new way of eliminating prion with all the advantages of a traditional small molecule. So these are small molecules that, as I said, can be taken in pill form and are also brain penetrant and allows us to lower PRP levels across the entire brain in a very even way. And this, I think, has many advantages over other approaches that are currently in development, which require invasive and painful intrathecal injections into the spine, etc. So switching gears now and talking a little bit about small molecule drug development. Just to give you a sense of the time frame of small molecule drug development, usually from an idea to approving a new drug takes on average 13 years. So it really is a very, very long process. And you can split up this process into different phases: the preclinical phase, the IND enabling phase, and then phase 1 and phase 2 or 3 clinical trials. And I've put above these roughly how long these different phases take. And in the preclinical phase, this is where GATE is right now with our molecule. We make and test thousands of different small molecules. And the aim is to make them more potent, make them more selective, to improve their pharmaceutical properties, and to test the best compounds in mouse model of prion disease. And as I said, I showed you a little bit of data. This is where GATE is right now. Now, once we've found the final molecule that we are interested in, we pick, we call this a development candidate. This is our very best compound that we think will become the final drug. We then spend around a year doing comprehensive toxicology testing. We want to make sure that these molecules are safe in animals, and we want to make sure that we can produce sufficient supply of the drug at a sufficient purity to support human dosing. And at this point, we're also preparing for our first in human clinical trials, including picking the doses that we want to test. And this takes around a year. After that, we enter what's called phase 1. And this is where we administer our drug to humans for the first time. And it's typical to start with a low dose and make sure it's safe before increasing the dose slightly. And it's called the dose escalation, and we go from there. And the aim of this phase 1 study is to confirm safety, to confirm tolerability, and to confirm that we are able to lower prion in humans in a safe way. And actually, if you've heard the news about Ionis, this is the phase in which they are right now. And once the drug has been deemed to be safe, once we can see robust prion lowering, then we will go to a phase 2 or 3 trial. And this is really the opportunity to show that your drug works to improve clinical signs or clinical symptoms. And this is where we will test the drug in patients. And approval usually requires a definitive clinical impact. And this is usually something like survival in the case of a very severe disease, such as prion disease. And so this is a little bit of an overview of the entire process, and hopefully gives you a sense of why it can take so long to go from the preclinical phase all the way to an approved drug that can be given to patients. So lastly, looking ahead to the clinic. So what can we as patient advocates, as families, as clinicians do to improve our odds of success? So I will break this section up into 3 quick bullet points. The first is to provide and advocate for historical clinical data. And what do I mean by this? If we can understand the natural course of the disease, this is going to help us design a very good, effective, and efficient clinical trial. So we need to be able to determine what a meaningful effect size would be on a drug. Are we really improving survival for patients? We need to determine how many patients we need to enroll in the trial to have the statistical significance we need to show a benefit. And we need to determine that if we should use a placebo control group or whether we can use what's called a natural history or synthetic control arm, i.e. we can use published data as the control instead. And to do all these things, we need to have a really good data set demonstrating the natural course of the disease. So what do we mean by historical data? What I mean is data on disease course from hundreds of different symptomatic patients. This is things such as time from first symptom onset to death, time of first suspicion of prion disease, time of probable diagnosis, clinical symptoms and how they vary over time, whether the patient is male or female, their codon 129 genotype, whether you're MM, MV, or VV, the age at diagnosis. This is just a few of examples. And this data can be, of course, fully anonymized. It does not need to be patient, have any patient identification. But having this type of data for hundreds of different patients is absolutely crucial for companies such as Gate to design the best possible clinical trials and to bring therapies to patients as quickly as possible. So if it's within your power, please help advocate for, and please help provide even historical clinical data to be published so that the entire field can use it. Of course, it's also very important to diagnose the disease fast and to raise awareness for when a trial is imminent or ongoing. The sooner you can diagnose a disease, the easier it will be to intervene. So once a trial is imminent, it will become very important to decrease the time from symptom onset to suspicion of prion disease and decrease the time from suspicion to a diagnosis, because this will enable us to consent patients fast and enroll them very quickly into clinical trials. And of course, all this is about is that the sooner we can lower prion levels after symptom onset, the higher the likelihood of a therapeutic benefit. And lastly, it's important, of course, to keep raising awareness and visibility of this disease. It is a rare disease, and it will take a village. It will take all of us working together in order to find a treatment for this devastating disease. And we all have our contributions to make, industry partners, patient advocates, families, clinicians around the world working together to educate people about this disease and working together in order to bring a therapeutic that's so desperately needed for patients and their families. So thank you very much for your attention today. And yeah, it's a real pleasure to be here speaking with you all.
Thank you so much, Nina. I couldn't have done it better in such a very, very strict time frame. I guess we all got a very good idea of what this new drug does for CJD or for prion diseases. Sorry. One thing that I guess I didn't get quite correct. So is this drug working only for sporadic, or is it also for familial?
It
Or,
will work for both familial and sporadic. So no matter the etiology, the therapeutic hypothesis is really if we can lower PRP levels, we can treat any of the etiologies. That's right.
yeah,
Yeah.
which is very, very good, because in some of the other approaches for therapeutics for prion, it was like strain specific, and as you say, this will work for any type of prions, which is also a very, very good approach. I don't know if we have some questions from the audience. I guess I have something in the chat over here.
Yeah, the first question is about kind of, is it safe to eliminate prion protein?
Right.
I think I addressed this a little bit in the talk, which is that there is evidence to suggest that although, yes, prion is involved in different cellular processes, it's likely safe to eliminate it. As I mentioned, there are cattle, goats, sheep, rats that lack PRP. and they seem to be normal. There are also humans that have one less copy of PRP, just naturally, and they also seem fine. However, we don't know, right? No one has eliminated PRP with a therapeutic before now. Ionis is the first to try it. And so there's no doubt that we will need to monitor this carefully in the clinic. And we also need to monitor it when we do our toxicology testing, as I mentioned in the section about drug discovery. So we'll definitely keep our eyes open for that. I think Eric, who's talking later, he can also, he likes talking about this topic, so he can give his answer as well. Yeah, and I think we need to bear in mind that prion disease is very devastating and rapidly progressing. So it's always about risk benefit, right? That there may be a risk to eliminating PRP, but how does that compare to the risk of dying from this disease? And that's, I think, the context in which we think about that. Yeah.
Thanks.
It's very exciting information right now.
Yeah. Yeah.
There is another question in there that says about what do you know about immunotherapy,
Immunotherapies.
and if there is any specific treatment also for Lewy body dementias.
Yeah. Unfortunately, not my area of expertise, so I feel like I'm not the best person to comment, unfortunately. But thank you for the question.
Okay, I guess we can leave that for Eric.
Yeah. Yeah, he may know more. Yeah.
Okay, so what are your perspectives about this treatment? When do you think it will be, let's say, ready to go into the how do you say it, over the counter to get this medication?
Yeah, great question. So as I mentioned, it's a pretty long process of drug discovery, and we are in that first preclinical phase. On average, it takes 13 years. You know, if we look at drug discovery from discovery or that first idea to approval. Approval means when the drug will be available to buy, right? So I can't comment on specific timelines, but we do hope to be at least testing the drug in people, so in the clinic, so in phase 1 trials, in a few years' time. So we can share some more specifics. I don't think we're ready to kind of commit to a specific time that will be in the clinic, but it'll be sooner that we'll be in clinical trials. Yeah.
Great. So whenever you're in the clinical trials, take a look into Mexico. We may be
100%. We will be in touch. Don't worry, Victor. We will be in touch.
Thank you so much.
Yeah.
Thanks so much. I don't know if there's any other question from the audience or even from the also speakers that are already connected with us, Susan, Dina, Beth, and Sarah, or Francisco. If you want to make or do any make any questions or any commentary to this great work that Nina is leading in the prion field. Oh.
Yeah. Yes. Yes. Hopefully, it should work in every etiology. Yes.
Yeah, so and as you can see, Nina is not that bad for understanding Espanol, and we some of us know the reason of that, but we're very happy that you can also be very fluent for the Latin community as well. Well, if there is no other question or commentary, I will say that we are very, very, very happy not only for having you, Nina, but also for that you are giving us hope for the families and for all the people affected with this kind of the diseases. And also, I know you collaborate a lot with Eric
Yeah,
Minkel, who will
that's
be
right.
also,
Yeah.
he will be
Right.
giving us his talk in an hour from
Great.
now.
He's gonna
So
do a great job. So
yes,
yeah.
as much as you did, I'm for sure. So I guess, Nina, that we will have to move on now to the
Thank
CJD
you for the
international.
opportunity. Yeah. And
Thank you so much, Nina.
to see you. Bye-bye.
Thank you very much, Nina.
Thank you so
Thank
much.
you. Nice to meet everyone. Nice to meet you, Francisco. Bye. Bye.
Bye. See you. Yeah, so now we will move on to this great alliance that Suzanne and Dina are leading right now. Suzanne Solvins, who is the founder and leader of the foundation in Australia, and I will read, Suzanne, if that's okay with you, the information that you have at the web page for CJD ISA. Is it okay? Okay, Suzanne Solvins received human pituitary hormones on a fertility program for 3 years in the 70s. In the 90s, she was informed that she was at increased risk of developing iatrogenic CJD, and then told that she had received a contaminated batch common with women who had died of iatrogenic CJD. Since then, Suzanne was a founding member of the CJD Support Group Network in 1993 and took on the role of director in 2004, which is the foundation in Australia. And she's also founding member and co-chair of the CJD International Support Alliance. Dina, if that's okay with you, I will read a brief extract of the information in this webpage. Dina Simpson is a representative for CJD Insight, which, like the CJD Support Group Network and the CJD Foundation in USA, is a founding member organization of the CJD International Support Alliance, which began in 2006. Dina is now a co-chair of the CJD International Support Alliance, along with Suzanne Solvins. And Dina is a member of a genetic form of CJD, which is running in her family. She has lost 13 family members in spanning 5 generations. Beth Marsh, and I found your data. I googled your data, Beth. I know you are the CJD support network, and you took the leading role at the CJD support network in November 2021. We are very, very happy to have you, as well Sara Gonzalez. And I also Googled that. Sara Gonzalez is la vicepresidenta, vice president of the Spanish Foundation for Prionic Diseases, which I believe was founded by Dr. Castilla. Isn't that right? Yeah, I've been with Dr. Castilla's quite a few times at the meetings, and I know that he's also been doing a very good job of establishing the foundation in Spain. So I will leave the microphone to all of you. I will first start with Suzanne and Dina, who are the co-chairs of the CJD International Support Alliance. Once again, thank you so much for being with us and for giving us the opportunity to listen, know, and try to get involved in the efforts that all of the families are doing for the wellness of all of the patients with CJD. Thank you so much, and Suzanne and Dina.
Are you putting up our slides, Victor?
I can do that. Do you want me to put the slides?
Yeah, I thought you were doing that. I can do it if not.
Whichever is better for you. I have them ready, but if you want to...
Yep, do that.
Okay. That. Okay, so one moment.
No, I can share. Let me try.
Yes, please, because I thought I had it ready, but now it's... Sorry. Okay.
Okay. We just have to see if the sound's going to work.
It is. We can hear you and we can see the slides.
Okay. Well, thank you for inviting us, Victor, to bring the voices and faces of those personally affected to members of the Mexican Prion Disease Foundation. As you've introduced us, and as you say, I am the director of the CJD Support Group Network in Australia, and I'm also a director of the International CJD Alliance. Dina, would you just like to introduce yourself?
Yeah, good afternoon, everyone, and thank you for having us. And as Victor said, I'm the director of CJD Insight in the USA, focused on genetic prion disease. Because I do come from a genetic family, and I am co-director of the Alliance with Suzanne.
And today I will also introduce representatives from two member associations of the Alliance, Sarah from Spain and Beth from the UK. Now we would like to introduce the members of the CJD International Alliance.
You can't hear it.
Can't hear it. Could that be heard? Okay.
No.
Do you want me, Susan, to try to show the videos? I don't know if that will
Yeah,
work.
you probably will have to, Victor, because we spent a lot of time trying to sort this out, and I'm not sure.
Okay, let me just try to. 1 minute. So I will share. So can you hear them? No.
No. Okay, hang on. I'm not sure, Victor, because I can't get into the part where it says share the so I'm not sure that.
Can you hear that?
No.
No. Okay, so I will stop sharing. So maybe you can share with your computer.
When you
Doctor, si
share,
está
Suzanne, does it give
compartiendo.
you the option to select share sound?
Yeah, it does, but I can't get into that section. That's what we tried last night. Oh. Here we are. Here we are. I've got it now. Okay. Okay. I will just go back and see if it's working. I think we'll have to keep going. All right. So we have.
Suzanne, you need to reshare.
Oh, okay. Sorry, I'm into the. Okay. Working now?
Yes.
Okay, We can hear.
Okay, good. We now have 12 dedicated member associations, 6 associate member associations and 1 country contact, which form the Alliance. In 2006, the CJD International Support Alliance was first established at a CJD Foundation family conference in Washington, D.C., founded by a very small group of patient associations. To focus, the focus of the CJD ISA is to continue to assist prion disease associations globally to provide support and assistance for all affected by prion disease. In 2023, the Alliance became a registered not-for-profit company. In 2024, we achieved charity status. So what changed in 2023? The Alliance received a grant from Ionis Pharmaceuticals, and this grant provided the funds we needed to become a legal entity. From 2006 to 2023, the work of the Alliance was all done on a voluntary basis. As a not-for-profit company, we have 3 directors, Professor Richard Knight, Dina Simpson, and myself. Our board consists of some very familiar faces, Professor Brian Appleby, Professor Michael Gueshwin from the USA, Dr. Joaquin Castella from Spain, and Beth Marsh from the UK. The availability of funding has also seen the introduction of our grant program to assist prion disease patient associations. Our funding priority is to assist associate members to become established by funding the following: development of a logo, the cost of setting up legal entity, charity status in their own country, development of a helpline, development of a website. And as you know, Victor, it took a long time for the Mexican Association to develop from an associate member to a full member. So this is very important funding to help to save that long period of time. So followed by funding support for member associations to assist them to expand their services by funding a startup meeting for the CJD community in their own country, assistance to develop resources with information on prion disease for patient care in their own language, to help them to maintain and update a website. Why are patient associations so important? Prion disease is rare. It can be extremely rapid, leaving patients and families feeling isolated and overwhelmed. Prion disease is always fatal, is transmissible, and can be genetic, making it unique and a devastating diagnosis. Each association is required to have a medical director to assist with clinical questions, and often these clinicians can review MRIs for the families and share knowledge with the clinical team caring for a patient. Information about testing procedures, patient care and options vary from country to country, so local knowledge is vital. For family members, being able to share their personal story with others in their own language and country is often a lifeline. Fundraising empowers the families and the CJD community to feel they can help and do something after dealing with an illness that they left their loved one without any option of treatment or cure. And we hope that's changing with what we're hearing today. Being part of a prion disease community that is united and working together to promote awareness and help research is very healing. So the future for the Alliance: we will continue to apply for more grants. We'll launch round 2 of the Advocacy Grant Program, support patients, families and caregivers to develop resources, continue collaborating with companies working towards a treatment or a cure. And I think, as Nina made very clear at the Prion conference, this is very, very important, this collaboration with the Alliance and the member associations and the CJD community when you're looking at treatment trials. We are very proud to have the Mexican Association as a member of the Alliance. We have just completed our 16th presentation at an annual Prion conference in Nanchang just a week ago. We would also like to share the work and achievement of 3 patient associations, which demonstrates the importance of establishing associations in more countries. So to start, I'll introduce the committee of the CJD Support Group Network in Australia, wearing our new United for Prion Awareness T-shirts that we're all going to wear on the 12th of November for International CJD Awareness Day. As Victor has mentioned, I actually am at risk of iatrogenic CJD from receiving human pituitary hormones, and our network was started in 1993, and we are fortunate that because of the cohort at-risk patients, we did achieve funding from the Health Department, and this funding does continue today. We've been very lucky that our contract has been reviewed, and we are actually funded now only in recent years to support all Australians affected by prion disease. We have just hosted our 15th annual family conference in Melbourne, and we also host annually a meeting in each state of Australia. To expand on the work of our education program, we are now hosting clinical workshops targeted at educating neurologists and other clinicians, and we thank our experts for their assistance and support in this work. Our CJD community is dedicated to raising money for research into prion disease. Family donations help us to fund research projects, purchase diagnostic equipment, and that's the only reason we could get RT quick going in Australia was from family donations to purchase the equipment in the beginning, provide research scholarships, and our most recent donation is for year 1 funding of a longitudinal study. The network has to date contributed $2 million in funding. Not all associations are fortunate to have funding, so for new and smaller associations around the world, it will be really important, and some of the funds that they receive as donation will have to be used to expand on their services provided in addition to them supporting research in their country. We are also hoping that our grant program for members can continue to grow and contribute more assistance. And now I'd like to pass over to Sarah from Spain to talk a little bit about the Spanish Association. Thank you, Sarah.
Thank you very much. Hello, everybody. I'm very glad to be here. The Alliance thought that maybe it's much better if I speak in Spanish, so we can have the meaning or the speaking in 2 languages, okay? It's fine with everyone?
Yo creo que no hay problema en que
como decía, soy la vicepresidenta de la Fundación Española de Enfermedades Priónicas. Joaquín Castilla es el fundador y es el presidente y es el que, bueno, pues el pilar de la fundación, como todos sabéis, y todos los que conocéis a Joaquín Castilla. I think I have somebody can pass. Okay, thank you very much. Bueno, ¿cómo trabajamos? Bueno, la fundación nació en el 2013, por lo tanto llevamos más de 10 años trabajando por los derechos de todos los afectados, apoyando a los afectados y, bueno, pues intentando contribuir a la investigación con todo lo que recaudamos a través de eventos y donaciones. ¿Cómo trabajamos? Pues eso, todos somos voluntarios, no tenemos ningún asalariado. Los socios no tienen que pagar ninguna cuota y entonces lo que recaudamos podemos trabajar gracias a las donaciones de particulares y de empresas, a la venta de artículos y a todas las actividades y los eventos que realizamos siempre de carácter deportivo o cultural. Can we pass the slide? Okay, thank you. como decíamos, hacíamos gran cantidad de eventos distintos, siempre deportivos o culturales, y todo lo que intentamos es dar la máxima visibilidad a la fundación y a las enfermedades priónicas, intentando no solamente recaudar para investigar en fondos, sino para ser visibles para todos aquellos casos que vamos a tener en el futuro, que nos puedan contactar lo antes posible y poder darles apoyo desde el primer día, que es muy importante, que sabemos que ese apoyo psicológico que ofrecemos, ese asesoramiento científico, ese apoyo y seguimiento y acompañarles en el camino, siempre es mucho más eficiente si estamos desde el primer momento. Lo que estamos viendo son fotos de carteles y de eventos que estamos haciendo. Como veis, todos son donde captamos a distintos grupos de empresas, trabajadores y personas para llegar a, bueno, pues eso, al máximo público posible, para que todos tengan la información y podamos seguir ampliando. We can pass. Okay, thank you. Es muy importante para nosotros intentar resolver todas las dudas y todas las áreas vacías que estas enfermedades tienen y que sufren los familiares. Uno de ellos son, por ejemplo, el diagnóstico genético preimplantacional, que luchamos con las entidades gubernamentales y que asesoramos a todos los pacientes que quieran tener una descendencia libre de este gen. Nos enfocamos siempre a la búsqueda de una terapia y constantemente escuchamos las nuevas ideas o las líneas de investigación. Incluso hemos creado un email, ¿vale?, que está disponible en nuestra web para que cualquier investigador o cualquier científico pueda bueno, pues solicitarnos esa ayuda y poder colaborar científicamente con ellos para que se pueda llevar a cabo. La Fundación cada vez tiene más medios o tiene cada vez más hueco en los medios de comunicación, tanto en televisión como en radio como en prensa escrita. Y bueno, pues ya es cada vez más frecuente las apariciones que tenemos y en muchos casos son ellos los que nos llaman y nos demandan por información y para hacer artículos o documentales. O sea que el trabajo, ya se está viendo los frutos del trabajo que llevamos haciendo, pues son más de 10 años. Y lo de los medios es muy importante para poder dar visibilidad y poder obtener ayudas, claro. Este año, en mayo, recogimos el premio que nos otorgó la Sociedad Española de Neurología. Es un premio que reconoce nuestro trabajo, nuestra visibilidad, nuestro apoyo a la investigación y es un galardón muy valorado y bueno, pues dado por la Sociedad Española de Neurología, y es el premio de Alzheimer 2023. Como nosotros somos una gran familia, porque lo más importante, lo que más tenemos que resaltar en la Fundación Española es que tenemos una comunicación muy amplia y muy buena con todos los socios y los afectados, ya sean de carácter genético o esporádicos. Y entonces lo que conseguimos son tener donaciones tanto de orina como de sangre regularmente. Cada 6 meses tenemos más de 125 donantes y entonces eso lo que nos da y las consecuencias que tenemos es que podemos trabajar y podemos dar a las investigaciones material para que puedan seguir investigando y poder avanzando. Pero es una cantidad que nos ha llevado mucho trabajo, pero la verdad es que son, pues eso, más de 125 donantes asegurados que cada 6 meses nos dan unas muestras de orina y de sangre. Y muchísimas gracias por vuestra atención. Os paso con Beth. Beth, it's your time. Thank you.
Thank you, Sarah. Thank you. So hi everyone. As I've already been introduced, my name's Beth. And so I'm here because I am the coordinator of the CJD support network in the UK And also because I'm a daughter. So in 2016, my father died with sporadic CJD 6 weeks after he was diagnosed. Thank you. Next slide, please. Sorry. So every family's journey is as unique as they are, of course, but there are certainly, I think, commonalities in the experience of prion disease, and these are reflected in this word cloud. These are responses that I received from members of our UK community who are affected by and at risk of all different forms of the disease. When I asked them to give 3 words to describe their experience. And that brings me on to the CJD Support Network. So the network was set up in 1995 in the UK and achieved independent charity status in 2003. And so it has worked for nearly 3 decades now to help alleviate the confusion, isolation and devastation that can be experienced by those in our community. In my role, I maintain strong links with specialist clinicians from Edinburgh and London who do so much to support the families in the UK, and some of whom are pictured here sitting alongside family members as part of our fantastic executive committee. And so as a well-established organisation with a robust governance structure and a clear vision, the UK network has developed over time to provide information, support and help connect people through various means, so written fact sheets and newsletters, video resources, a helpline, caring grants and regular online peer support groups. We've also more recently expanded into providing research grants, which have funded a range of projects, including those related to diagnosis, prognosis, and most recently a pilot study, which is investigating a group psychological support intervention for people living at risk of inherited prion disease, which I think is much needed. And I'm pleased to say began recruiting in late September. Thank you. So our annual flagship in-person event, like many of the associations, I think, is our family support meeting, our annual meeting. And so reflecting the value of the work that can be done through these associations, attendees of our meetings, some feedback on the slide there, find it hugely beneficial to have the opportunity to meet one another, to connect and support each other, as well as to learn from and ask questions of national experts in the disease. The CJD Support Network initially received some statutory funding, but it now, and for a long time now, has relied entirely on the generosity of our members, donors, and the fantastic fundraisers within our community who do all sorts of things to raise money for us and remember people that they care about. So walk, run, swim, skydive, as you can see there, host events and much more. And I know, as Suzanne's already mentioned, that many of the other members of the International Support Alliance also receive no formal funding, particularly associate members, which is again why it's really important that the grants of the CJD International Support Alliance are available for helping these organisations to get established. Thank you. So alongside grief, there is hope in our community. You may have noticed the word hopeful in that word cloud that was shared a few slides ago. And it's really the work of the researchers, scientists, clinicians, people like Nina, who dedicate their time and effort to this field, which supports this hope. So I don't know how many researchers are in the audience, but this is just a few messages from our network to say thank you. And I believe I'll be now passing over to Dina.
Yeah, thank you, Beth. We now want to turn your focus to the Alliance's plans for marking International CJD Awareness Day, which is coming up on November 12th. November 12th was identified as International CJD Awareness Day approximately 15 years ago in the UK, and for more than a decade, November 12th has been honored and promoted around the globe. Our intention is to bring global prion disease support community together under the message that we are globally united for prion disease awareness. As part of this, we have developed resources that will be provided to members of the Alliance, our friends and advisors, and our collaborators to share with their communities. All resources are available on our website at www.r/cjdisa.com, and we'll share some examples with you today. Next slide, Suzanne. So this is just an example. There's many out there on our website of an artifact for Instagram. with our logo of Globally United. Here's another example of a format that's different. Next slide. We have similar for Facebook, so you'll see those out there. Next. Social media artifacts, as you see, we're using a background here on LinkedIn that we invite all of you to use over the next week or so as we approach November 12th. Next. Next. We also have out there a poster that can be printed. So we wanted to provide something physical that hospitals or clinician offices or even in your own organizations can have a physical presence and visibility to this important day. So feel free to use that. Next. As we post stuff on social media, we can insert any of those artifacts that are available on our website. We also encourage people to use 2 hashtags, hashtag CJD awareness and hashtag prion disease awareness. We want to include prion disease awareness because we want to promote and make sure we're inclusive of our GSS and FFI patients and family members. So we're promoting both of those hashtags. Next. So we reached out to our community and asked our alliance members and our associate members to collaborate on creation of a video that we can share leading up to and on November 12th. And we're so pleased to say that every member, associate member, and family contact responded and contributed to the video. We'd like to share that video with you today.
Globally united for prion disease awareness.
Unis dans le monde, sensibilisons aux maladies à prion.
Globally united for prion disease awareness.
We're globally united for prion disease awareness.
Todos unidos para crear conciencia sobre las enfermedades priónicas.
Weltweit vereint für das Verständnis von Prionenkrankheiten.
Globalmente unidos para sensibilizar por las enfermedades por priones.
Uniti in tutto il mondo per sensibilizzare sulle malattie da prioni.
Globally united, prion disease awareness.
Unidos internacionalmente pela conscientização sobre as doenças priônicas.
Globalmente unidos pela sensibilização para as doenças priónicas.
Wereldwijd verenigd om prionziekten onder de aandacht te brengen.
Globally united for prion disease awareness.
हम आर्मी सतह पर प्रायन की बीमारी की तहकीक के लिए मुतहद हैं.
Globally united for prion disease awareness.
Weltweit vereint zum Bewusstsein von Prionerkrankung.
Globalmente unidos por la concientización sobre las enfermedades priónicas.
So I think you could see that we had them do it in their own language. We had them do that in their own language. It certainly demonstrates the. You got to go to the next slide, Suzanne.
Okay.
We had them do it in their own language, and it certainly demonstrates that prion disease has no borders. It impacts everyone around the globe. Lastly, we want to acknowledge our friends and advisors. These are experts in the field of prion disease and consist of researchers, clinicians, academia, and they assist us in supporting patients and families and caregivers and all those at risk of developing prion disease. They've been especially helpful in countries where a support organization does not exist. Our friends and advisors currently represent 22 countries. Having said that, after our presentation and at Prion 2024 in China, several attendees approached Suzanne asking to become friends and advisors, including some countries that we currently do not have represented. So I think it demonstrates the importance and the impact that sharing and bringing awareness can have. It just embraces and expands our community to better serve our patients and families. We couldn't do what we do without their support. So just a quick video acknowledging our friends and advisors. So our final ask is that you join us in promoting awareness on November 12th and unite with us in support of pre-disease awareness.
Sarah?
Yes. Pues es todos unidos globalmente para crear conciencia de las enfermedades priónicas.
Thank you.
Thank you.
Muchas gracias por la presentación. Thank you so much for this tremendous presentation on showing how working together as a one major entity, major worldwide entity, we can do much and a lot for the people with prion diseases. Unidos globalmente por las enfermedades por priones. I remember when I was filming myself, I was wearing one of those CJD t-shirts, and then something came up, and then something came up, and then when I finally sent the video, Suzanne told me that something came up again, so I had to change it again. But I guess we'll have the same feeling in our hearts to help all of the patients and all of the families who are struggling with this disease in their families. And we in Mexico, with the leadership of Francisco Mota, as the president of the Mexican Foundation, we're also doing our part and trying to help the families, not only in Mexico but also in Latin America. We've also had some cases, some families to approach either us or any of you or in the world, asking for someone in Latin America, for someone to give them some information and help, and you or you always refer them to us. So we're also happy to assist them in Latin America, in any of the countries where there are no foundations or clinicians that we know of attending CJD patients. And thank you again for being with us, Beth, Sarah, Dina, and Suzanne in this day. And we will also be raising awareness. I teach CJD when I'm teaching with my med students at the university. So I try to make sure that at least one generation or one group of the bigger generation of the university know that CJD exists, and there's people that are already working as one for the good of these families. Thank you so much.
Thank you.
Gina.
Okay.
Is there Dina and Beth and Sarah and Suzanne? I need to be thanks with you. It's an honor to be part of this great movement. Is an honor, believe me. Thank you very much.
Thank you. We couldn't do it without all of you. So keep up the great work, and we'll do this together.
Thank you very much. Thank you.
Thank you.
Thank you so much. Well, we'll have to move on now to our next speaker, who's been here in the past minutes. He's also part of the CJD International Support Alliance. He's a friend and advisor, and he's neurologist. He went through his medical career at Albert Einstein College of Medicine, where he received his MD and PhD in neuroscience. He completed his internal medicine internship at UCLA and his neurology residency at Johns Hopkins School of Medicine, and has a fellowship in neurobehavior at the University of California in San Francisco Medical Center, Memory and Aging Center, where he stayed on as faculty. Es un honor poder contar con nosotros con el doctor Michael Geschwind, quien, como ya comenté, es médico internista y neurólogo que obtuvo sus grados en la escuela de Albert Einstein, en la UCLA y también en la escuela de Johns Hopkins. Actualmente es médico, neurólogo y parte del Centro de Envejecimiento y la Memoria de la Universidad de California en San Francisco. Y será un honor escuchar su presentación sobre un resumen clínico de las enfermedades por priones. It will be an honor to hear Dr. Michael Geschwin on the topic of the clinical summary of human prion diseases. Michael, thanks so much.
Great. Thank you, Victor, for the kind invitation. And this is a talk that I'd given a year ago, and I've just updated it a little bit. But this is, Victor asked me to talk about kind of the clinical aspects of human prion diseases. And I hope I'm not overlapping too much with Professor Zerr and Knight's talks, and I think they've already spoken. And then I know Eric will speak about some of the therapies, but hopefully I won't have much overlap. I apologize if there is. So I was asked to kind of give a clinical approach, a clinical overview of prion diseases, and the people can see my slides okay, is that right?
Yes, we
thumbs
we
up.
can see your slides and we can hear you very well.
Okay, good. Sorry. So typically we divide human prion diseases into three types: sporadic, acquired, and genetic prion disease. And sporadic usually is sporadic CJD. There are there's another subtype that's a subtype of sporadic disease that's not quite CJD. It's a prionopathy, or a disorder of the prion protein, in which the prion protein is sensitive to degradation by proteases. And particularly the Case Western group, Dr. Gambetti and colleagues is the first one to describe that. And that was, they call that variable protease sensitive prionopathy. But for the most part, it's very rare, only maybe a few dozen cases at most ever described. So I'm going to talk mostly about sporadic CJD and some classic and non-classic forms, and some issues with diagnosis. And then I'll talk also about acquired prion diseases relatively briefly. And then I'll end with some talk about genetic prion diseases and some research I'm doing on that, including collaboration with several of the people who are on this call, other researchers. So human prion diseases, sporadic, about 85 to 90% are sporadic or spontaneous. Most of these are, we call CJD or sporadic CJD or SCJD. And depending on the cohort, 85 to 90% of most cohorts are sporadic. The genetic are about 10 to 15% of most cohorts. A beautiful study, somewhat old, but a 2004 study from Professor Kovacs, who's now in Canada, but was in Austria at the time. He looked at the European patients referred to mostly European and Canadian centers, and I think Australia, who ultimately had genetic prion disease. And it turns out that 47% of them, almost 50%, had no family history of known prion disease. And in some cases, it probably was because the disease was there, but it was misdiagnosed as Alzheimer's, Parkinson's, or other related neurodegenerative diseases. Sometimes it was hidden from families. That's not uncommon, where one branch of the family knows the disease is there, but they don't want to tell other people, even other members of the family. Historically, these genetic prion diseases, which are caused by an autosomal dominant mutation in the gene that makes the prion protein gene. And historically, they've been divided into three subtypes: familial CJD, which there are probably a few dozen mutations that cause this; Gerstmann-Strauss-Scheinker, GSS, just more than a dozen mutations that can present like this disease; fatal familial insomnia, which is there's one mutation that can present with this disease. And that's kind of an antiquated way because now we know about octopeptide repeat insertions, which can present like GSS or FCJD or somewhere in between, plus stop codon mutations, which can look nothing like any of these. They can often have very early onset. They can last for years, if not even decades. And they tend to be a tauopathy as well as a prionopathy, so other proteins are involved. And then acquired CJD, even though they're the most notorious, they're less than 1% of all cases. Kuru from the ethnic or linguistic Foray group in Papua New Guinea, this disease occurred through endocannibalism, in which to honor deceased relatives, they had a mortuary feast in which they cooked partially and then ate their deceased relatives to honor them and to keep their spirit in themselves. And unfortunately, somewhere along the way, one of the people who died had CJD, probably a sporadic form. And then variant CJD, which is essentially due to exposure to BSE, bovine spongiform encephalopathy, or mad cow disease. That's usually how this occurred, and is usually through consumption of contaminated beef. But now there have been several cases occurred through blood donation and also through accidental laboratory transfer. And then here's a diagram of what our cohort looks like of patients with rapidly progressive dementia, or RPD, who had known or suspected prion disease. So most of these patients, not all, were referred to us as CJD, and we found that about 45% did not have CJD. And some of these in blue actually were referred to us even for our treatment trial for prion disease and for our diagnostic studies for prion disease. So they've been misdiagnosed. And some of these CJD really wasn't in the differential, but they had a rapidly progressive dementia. Of our cohort, this is as of about 3 years ago, we had 900 evaluated. Now we're closer to 1,000 and probably closer to 3,500 referrals. And we found that about a third had sporadic CJD, and then about 14% had genetic. A small percent we don't have their mutation status. And these, by the way, include people who are mutation carriers, most of whom will eventually develop prion disease, but some of whom are just carriers and they have a mutation that's not fully penetrant, and they hopefully some of them will never develop prion disease. And that's kind of the work that a lot of researchers around the world are trying to figure out. Are people who have mutations, is there a way to predict who will ultimately develop disease, particularly for mutations that aren't 100% or close to 100% penetrant? And then acquired prion disease in our cohort is less than 1%. And these include primarily people who got exposure through accidental medical procedures or variant CJD. I think I had another figure, but it's missing here. So for sporadic CJD, the peak age of onset is unimodal. And it's a high peak in the mid 60s. That's the most common, mid to late 60s, depending on the study. And it really drops off pretty precipitously after that, meaning that it's unusual to get the very old or the very young. But the youngest sporadic case I'm aware of is a 12-year-old from Spain and a 96-year-old, I think, from Italy with sporadic CJD. Median survival in the literature is about 5 to 8 months. In our cohort, it's about double that. And that's because we spend a lot of time trying to identify the first symptom, whereas most of this survival data is from medical record review, from autopsy studies. And we have the benefit of spending several days with the patients and families. And so we can actually really try to get, we usually find that sorry, I'm going to turn down my phone. We usually find that the onset of the disease was much sooner than earlier than the medical records had stated. It's not unusual for it to be 6 months earlier. We'll find something abnormal that we think was actually the onset of the disease that maybe family members or clinicians hadn't paid attention to. But probably 85% of patients or so live less than a year. The incidence is tends to be about 1.2 to 1.4 in most developing countries. But based on data from the UK and from the US, the CDC, this is the number of cases worldwide. We'd probably expect there to be 400 to 500 cases in the US per year, maybe a little bit more. And then in Mexico, we'd expect this amount with these numbers. But the actual person's lifetime risk of dying from CJD is actually about 1 in 6,000. And that's based on, if you look at most people in the developing world will live through the peak age of CJD. And if you look at all deaths from CJD over all deaths and only looking at the people who live through the peak age, that's about a person's lifetime risk of dying from CJD is about 1 in 6,000. So here's a typical patient. Just because of time, I'm not showing videos because those take a lot of time. But this is a 61-year-old who was referred to our clinical treatment trial. When he came to see us, he had had 3 months of symptoms, and his symptoms started with confusion, forgetfulness, vision changes, and word finding. So he became confused in his house. He became disoriented about how to get from room to room. He began to have trouble reading. At work, he used a computer, and he came home and he said to his wife, "Honey, I can see the words on the screen, but I no longer know what the words mean." So he was having a problem with reading or alexia. He's also having problems with writing agraphia, and those we know where that occurs in the brain. He was also having something called visual distortion or macropsia, where if he looked at his hands and feet, he said, or sometimes even other people's hands and feet, they seem grossly enlarged. And that's a phenomenon that usually occurs in the right temporal parieto-occipital junction in this part of the brain. And as we'll show you, his MRI showed abnormalities in all these areas. By month 2, he started repeating what others said. He had decreased speech output. He was an active bicyclist. He stopped biking. He couldn't he had trouble controlling his balance. He couldn't figure out how to use utensils for eating. He was unable to help with the household chores that he used to actively participate in. By the time we saw him, he could no longer be left alone. He had rigidity of the right side of his body, and he could walk, but very slowly. And he had trouble getting words out, trouble having conversation. This is a series of, this is one of his MRIs when we saw him. The white areas here are abnormal, and the black areas here, black on gray, where the arrows are pointing to, are the abnormal areas. And in this patient, most of the areas are in the cortex, the surface of the brain, and where it's bright on the diffusion weighted image, the DWI, there's corresponding darkness on the ADC map. And that tells us there's reduced diffusion of water. This is the same sequence we used for detecting recent strokes that have happened within about 2 weeks. Except in this disease, the pattern is not a pattern of blood vessels, of blood vessel distribution. It's a completely different pattern. And we've done some research showing that the pattern seems to affect areas of the brain called the default mode network, and that there are certain parts of the brain that are preferentially affected. And some of our research has also shown that the areas that are most affected are where the prion protein is most highly expressed in the brain. So it's not completely random. And in fact, colleagues around the world in Italy, the US, Germany, they've shown that the pattern of MRI can actually link to the subtype of prion disease and can help us, which can sometimes be helpful for prognosticating what a patient's disease course and survival would be, might be helpful for stratifying patients in treatment trials. Now, that was a patient who only lived about 4 months. He died after about 4 months. This is a patient who was 66 years old. And by the time he came to see me and my colleague at an adjoining university, Stanford University, he had 2 years of progressive language problems, or what we call aphasia in neurology. Without language is aphasia. He had word finding difficulty. He had trouble naming things. We call that anomia. He had trouble understanding long, complex sentences, particularly if they were on the telephone. But he was still very fluent. I'm not showing you the video, but he was able to give his entire history. When you asked him to name things, he was pretty good with naming common objects, but uncommon objects, let's say like a hammock, he had great difficulty naming. At the point we saw him, he was still mountain biking, kayaking, and driving a truck for a living every day. And this was his MRI. Again, you're seeing on this diffusion weighted image, abnormally bright areas here in the left tail of the hippocampus, left insula, the left side of the brain where language is, and then also in the right side of the brain too. And also in the back of the brain, this is a limbic structure, part of the brain that's a part of memory and emotion. So we diagnosed him with sporadic CJD. He lived for another year and then suddenly had a 2-week sudden decline and passed away. And autopsy showed CJD, even though he had a 3-year course. And he was driving up until 2 weeks before death. We'd taken away his license, but he still was able to drive. So we published him in archives are now known JAMA Neurology. So where the prions are depositing and causing pathological change seems to correlate with the types of symptoms that people will get. So this is the frontal lobe, the parietal lobe, the occipital lobe, the cerebellum, the temporal lobe, and each of these parts of the brain subserve different functions. And I've listed some of them here. So if the temporal parietal lobes are affected, you might have an aphasia disorder, or if it's on the right parietal, you might have visual spatial dysfunction. If it's here in the vision, where vision is processed, you might have blindness or difficulty seeing things. And it may not be the actual part of the brain that's involved, but it might be the pathways connecting them that get involved in this disease as well. We've shown that about 2/3 patients in our cohort at UCSF have a pattern of MRI involvement with the cortex and the deep nuclei involved, and about 1/3 predominantly only have the cortex involved, and then about 2 to 5% only have subcortical involvement involved. And other laboratories have shown very nicely that some of these MRI patterns, again, can subtype the type of sporadic CJD and even help us. And that by subtyping it, we know what symptoms we might expect in the future and also what the average survival might be. We can't predict at an individual level yet, but we can give a gross approximation. And these MRI changes we've shown to be a very highly sensitive and pretty darn specific for prion disease, but they have to be read carefully. We've also shown, I'm not going to show you the data, that a lot of radiologists around the United States miss the diagnosis. And so we really have to, we spend a lot of time training physicians, neurologists, people who might see patients to read their own images and know how to interpret them for CJD. Acquired human prion diseases, I've mentioned Kuru. I didn't talk much yet about the iatrogenic. This is medically induced. And there are patients who've had human pituitary hormones, including human growth hormone most commonly, but also hormones used to help women get pregnant or women who have infertility issues. These are hormones that are derived from the pituitary gland from cadavers, patients who've died. And unfortunately, there have been some patients who died and parts of the brain, either from the pituitary or the dura mater, the covering of the brain, or the cornea of the eye, have been used, taken out of patients who've died, who unknowingly had prion disease, and then those hormones or dura mater or cornea were used in other patients. And unfortunately, it transmitted the disease to those patients. And usually it took, in most cases, it took years. Sometimes it was less than that. And then even there have been EEG depth electrodes that have been implicated or actually proven to be, they've used them in one patient to measure their seizures. They cleaned and sterilized them in a way to remove viruses and bacteria, but not in a way to denature or remove or inactivate all proteins. And unfortunately, those electrodes were then used in other patients who later developed CJD. And then they still have those electrodes, they put them into an animal, and the animal developed prion disease. And then the variant CJD, again, I already mentioned that as being due to exposure to bovine spongiform encephalopathy. CRU, I already mentioned a little bit about this disease from the Foray linguistic group in Papua New Guinea. It means to shake or tremble. And here's a young boy, usually affected women, women and young and children in the population. It's probably because they were, when they participated in these ritualistic, cannibalistic um, feasts, mortuary feasts, they tended to get the brain, the spinal cord, the less desirable part of the feast, which was the meat, the muscle, which had fewer prions in it. And so they were probably more, they had a higher load of prions than they were more likely to get it. At peak of the disease, it killed about almost 1 to 2% of the population. And it was much more common in women than men. Again, it's probably because of how they participated and what parts of the deceased body that they ate. And then less than 20 years of age was about 20% of cases. And again, women more than men, and particularly with the older. And Dr. Collinge's group in London has shown that the incubation period, even, gosh, more than a decade ago, was greater than 50 years. So this Karoo died out when the missionaries stopped the endocannibalism practice, but even years later, there were rare cases that occurred. And here's another example, Carlton Gajusek and Professor Vigas from Australia evaluating a poor boy who was suffering. So if we look worldwide, it's an old paper, but it's still pretty accurate. And that's looking at iatrogenic or inadvertently caused by medical procedures. And the most common have been dermatograft and human growth hormone, less commonly gonadotropins, again, often used for fertility. And then there have been some cases of variant CJD through blood products. And then I mentioned some of the other cases. And the incubation period, you can see here, 12 years, but the wide range. I had a patient who walked into my emergency room who had growth hormone. He was 33, had growth hormone kind of between the ages of 4 and 11. And we think he had at least a 22-year incubation period with iatrogenic CJD. What about variant CJD, the type that's most commonly from exposure to BSE? Well, we had a 20-year-old woman who came to us. She was in an elite part of the British military. Her first symptom were personality changes. She used to be very outgoing, and she started to become depressed, more withdrawn. By October through Christmas, she became so depressed, she didn't spend her vacation with her friends over Christmas. She stayed in her room during the Christmas holiday, was found banging her head against the wall. She was put into a psychiatric hospital and given a discharge from the military. But a month later, she started to have neurological symptoms, ataxia or uncoordination, chorea, which are sudden ballistic movements, usually of the limbs or different parts of the body. And then she came to our center in July, and we did, I think we did tonsil, maybe, I can't remember if we did brain biopsy too. We confirmed that it was variant CJD. We didn't have some of the diagnostic tests that are as accessible today. She went home on our treatment drug of quinacrine on open label, and we were told by the British press that she'd recovered, it was a miracle. Unfortunately, she came back 2 months later, and it was clear that there was unfortunately no miracle. She had worsened in many levels, and she passed away, sadly, just a little over a year after onset, about 14, 15 months after onset. This was her MRI, and she has something which we call the pulvinar sign, which the posterior part of the thalamus is brighter than the anterior putamen. This is usually on T2 weighted images. It probably holds for diffusion weighted images, but the study, to my knowledge, has never been done to show that, just because these images weren't as available in the UK during the epidemic as they were in the United States and some other countries. but that's the pulvinar sign, and she had it. This is not diagnostic for variant CJD. We can see this in sporadic and genetic, but it certainly would make us very suspicious for variant CJD. How is it different? Well, as I mentioned, it's linked to BSE. It's also patients are younger. Instead of a mean age of about 68, median age, it's 28, so it's 40 years younger. There's a wide range of onset, but the vast majority, all but a handful of patients, have been less than 55. Instead of about a 5 to 8 month survival, it's about 14 and a half months. The symptoms overlap, but there are some differences. They often have a very prominent early psychiatric prodrome, and then often have a migrating pain syndrome. But this doesn't rule out other forms of CJD that Dr. Appleby in our lab have shown that psychiatric symptoms are very common in even sporadic CJD. Sorry, my talk's on automatic and I'm not sure how to turn it off. So, and then the EEG and the MRI are somewhat different, as I've shown you. There's a different genetic susceptibility. The pathology is very different. And also where the prions are present in the body. They're very present not only in the central nervous system in the brain, but also in the lymphoid tissue along the gut. And that's probably why this disease is much more transmissible. In the Western Hemisphere, there have only been 6 cases, 4 in the US and 2 in Canada. No cases have been acquired in North America. All of these cases, we have good evidence as to that they are being acquired outside of North America. And then the 2 most recent cases in France were believed they occurred in laboratory workers who were working with variant CJD models of mice. They were mice that had the human prion protein gene in them. And unfortunately, the investigators weren't, in some cases, might not have been following appropriate protocols, but regardless, they accidentally stuck themselves with needles from or sharp instruments that were used on the animals. So this is a graph of the BSE, or bovine spongiform encephalopathy epidemic, which really peaked in the kind of in the early 90s in the UK. And then the first case of variant CJD was noticed in 1995, late 94, published in 96. And this was kind of a map of the variant CJD cases in the UK. And you can see that there was a temporal relationship, a delay between one peak and the other peak of just over a year. And this graph from a very nice review paper by Inga Zer and some other people on the call in Nature Review Neurology, in which they looked at all the variant CJD cases in light blue or UK, France in the this reddish color, rest of Europe, and then the rest of the world. And these are the 2 more recent French cases that I'm aware of that were likely laboratory based. And to my knowledge, there have been about 238 cases of variant CJD ever diagnosed. We don't know if there will be another resurgence of this epidemic. Our hope is that there won't be, but they looked in the UK, and based on data from the UK, it looks like 1 in 2,000 people in the United Kingdom are walking around with variant CJD prions in their lympho-reticular tissue, in either their appendix tissue or their tonsillar tissue. And we don't know if those patients will ever develop prion disease themselves, if they can transmit the disease, or are they silent carriers and they'll never develop or transmit the disease. These are unknowns, to my knowledge. So here's a schematic. I'm going to switch gears now, talk about genetic prion disease. Here's a schematic of the prion protein gene. In red here are some of the mutations. In blue are some of the novel mutations that we found in patients at our center. Most of the testing was done through Case Western, the National Prion Disease Pathology Surveillance Center, who we tried to do most of our genetic testing through. And then below in green are polymorphisms or variants that seem to affect how the disease presents. this codon 129, if you're homozygous, you have two copies of every gene, so you can have two copies of this codon 129, and you can either be methionine or valine. And if you're methionine, or valine, it seems to increase your risk of prion disease and being heterozygous, so having a methionine on one allele, maybe the one you got from your father, and a valine from your other allele that you maybe got from your mother, that seems to be slightly protective against prion disease. And then looking at the London group, which Dr. Collins, Simon Mead, and others, they actually looked at people who survived the Kru epidemic in Papua New Guinea, and they looked at, I think, primarily women who survived the epidemic, and it turns out that they found that they overwhelmingly had a V instead of a G at this codon 127, which made them think that maybe it's protective against getting prion disease. So they made a mouse model, and to my knowledge, they still haven't been able to give it prion disease. So knowing this information might help us think about drugs and work that Dr. Minical and others are working on to try to find treatments for this disease might help us toward that. Here's another figure that we, in one of our chapters that in papers we've written, where some of the mutations can present looking like CJD in red. Some look like GSS, shown somewhat in green, and then there are the stop codon mutations shown here in purple, and these are ones where a truncated version of the protein is made, and these patients present very differently than other cases of CJD. They have younger onset often. They have a much prolonged course. The biomarkers that we use for sporadic CJD usually are negative. The MRI, the RT-QuIC, all those things. Excuse me. And some of these that are outlined in yellow that have been called mutations, others have shown, including Eric and Minical and Sonia Vlab, have shown that they're probably just maybe very low penetrance mutations. And sometimes I even consider them almost as risk factors, but they seem to increase your risk. The most common mutation in the world is E200K. So at location 200, there's an amino acid substitution. It's the most common mutation worldwide. It's probably the most common in Mexico, as well as in our experience in the US. There are at least 4 geographical founders from around the world, meaning this mutation started at least 4 different places around the world, and then those people spread around the world. One was Mediterranean or Sephardic Spanish Jews, includes probably most of all of the Mexican E200K families. Most of these families have no idea, not all, but that their ancestry is of Spanish or Sephardic Jewish ancestry. Many of these families converted to Catholicism during the Inquisition, eventually made their way to the Americas, and kept the Catholicism. Some of these families, though, unknowingly still practice Jewish traditions, like on Friday nights, the Jewish Sabbath, lighting candles, but they often will do so in secret in the basement. And it's usually the grandmothers or great-grandmothers that were doing this tradition. And then Eastern European from Slovakia, there's a cohort from there, another cohort from Japan, and probably another Austrian-Hungarian and maybe an Italian cohort as well, founder effect. Yeah, I think that was, here are the 3 people, Gerstmann, Strausser, and Scheinker. I think Austrian, German, and Russian, if I recall. These are the first ones to describe what we now call GSS, which is it's a genetic disease that has certain pathology. You get these plaques of the prion protein. And if you didn't know that it was prion and you just stained for plaques, you could mistake it for Alzheimer's disease. And for many years, these patients were mistaken to have Alzheimer's disease. But when they identified the prion protein and put the prion antibodies onto these brains, those plaques weren't Alzheimer's proteins, they were prion proteins. This is two patients who we've followed at our center, a 23-year-old daughter who had problems with her cerebellum, coordination, and memory loss, and her father, who was 26 years older than her, more than double her age, and he developed behavioral and cerebellar problems, and they developed them at the exact same time. So she had a rough life. She was a heavy drug user, really didn't take care of her health. When she was 23, she started to develop excessive thirst, handwriting changes, and then had behavioral changes, movement disorder, coordination problems, memory problems, worsening movement problems. And we diagnosed her with probably the third most common form of GSS, I think, in the world, A117V. And then her father, same month, developed symptoms, but he presented very differently. He had disinhibition and irritability. And he lived for about 2 years. She lived for just over 3 years. And this is their family tree. In blue are people who never developed symptoms. In black are people who we diagnosed genetically or pathologically with GSS. And in red are the people who, based on their history, likely had GSS. So you can see it can affect men, it can affect women, and it occurs in multiple family branches. When patients come to our center for our research, it's usually a 2-day visit. We do a quantitative motor testing shown here. We do a structured neurologic exam. We do cognitive testing. We take different samples, including skin biopsy, and we do sleep study. We're beginning to do sleep studies in these patients. Sometimes they get genetics counseling or social work. We do a lot of questionnaires. And then if they're symptomatic, we talk about palliative care, end of life planning, hospice. We're pretty aggressive about talking about autopsy, particularly working with CDC and the National Prion Disease Pathology Surveillance Center to make sure that all of our patients who are willing to get autopsy get autopsy. And we have historically, I think, a 70 to 80% autopsy rate, which we think is pretty good, but we could do better. And then there are other studies that we're doing too to try to minimally invasively study prion disease. And one of my goals is if this is the clinical onset of genetic prion disease, we divide mutations into kind of CJD mutations or GSS. And we think that we know that the biomarkers are different, the imaging is different, the spinal fluid tests are different, and we think, and their course is different. So we think the biomarkers might be slightly different between the two. In dotted lines, we show what we think will be the trajectory before clinical onset for the slow prion disease. We think we might be able to detect changes years before onset. And then maybe for the fast ones, maybe we'll detect changes maybe closer to onset. But many of the labs that are studying genetic prion disease now have found at least one spinal fluid biomarker to be positive up to, in the E200K type of prion disease, to be positive even 5 years before onset. We have one patient who are CSF RT quick became positive 4 years before onset. And we followed her for 4 years, and unfortunately, after 4 years, she developed symptoms and then had a rapid course and passed away. And then other in Israel, in the UK, in Germany, Sonia and Eric and their cohort, we're all finding the same thing. I don't think we know enough yet if it's going to be true for everybody with E200K. One possibility is that the evidence seems to suggest that once you have a positive RT quick, that means that the disease has begun. There's still other possibilities. It might be that we're just detecting the disease earlier because the prions that are being made from the mutated allele are more susceptible to misfolding and causing a positive test. And that still remains to be seen, but our concern is that it really does mean the disease has begun. And these are people we might want to start treatment once we find a good treatment. So this is data that's over a year old now, but we followed almost 200 participants from 73 families. We bring in people from the families who are carriers of the mutation, but we're just as interested in the non-carriers because there are perfect control groups. And we're trying to bring in people who have fast type mutations like CJD, like E200K, and slower that present like GSF. Here are just some of the mutations that we've brought in. You can see it's the most common is E200K, then D178N, this is a disease that can either present as familial CJD or present as sporadic, as CJD. And then here we have some of the GSF type of mutations. And then for some patients, we only have 1 or 2 people who participated with that mutation. And then here's a similar graph, but dividing into people who are carriers in blue and non-carriers. And the orange are going to be our control subjects for much of our research. So prion diseases are incredibly diverse. The diagnosis can be difficult. And hopefully Dr. Zur has already spoken to you about that, and Dr. Knight and others. We think that genetic prion diseases might teach us a lot about how the disease begins and occurs and spreads in the brain, and might help us understand and treat the more common form of sporadic disease. And then these 2 bullet points I didn't really talk about, but we now know that this idea that a protein changing conformation and that misfolded, misconformed protein causing disease is not unique to CJD and related diseases, but it seems to be how Alzheimer's, Parkinson's, and other neurodegenerative diseases that are due to protein misfolding are also occurring. The distinction is that those other diseases, the misfolded proteins don't work quite as fast as they do in prion-related or PRP prion disease, and they're probably not as transmissible. So some people don't like to call these other diseases like Alzheimer's and Parkinson's prion disease because that has implications for infection control. Some people use the term prion-like. I don't think we have a great term yet for it. But the key is that the mechanism that was identified for causing prion disease is not unique to this disease. And it seems to be emblematic of many neurodegenerative diseases. And this realization over the past 10 years has just completely changed the field of how we think about and how we're going to treat neurodegenerative diseases. The work I've shown you has been very collaborative. I have a lot of colleagues at UCSF in my group at the Memory and Aging Center and the Neuroimmunology group and social medicine, and then a lot of collaborators from around the world. And most of my funding has been from the National Institute of Health. And I want to thank the patients, the families, the NPDSC. They do a lot of our genetic testing, a lot of our autopsies, the CDC for funding them, and the CJD Foundation, and the CJD International Support Alliance, who you just heard from, and the CJD Foundation as well in the US, and some other CJD foundations and patient organizations in the US. Thanks very much.
Thank you very much, Michael, for this great presentation where you've shown us not only the clinical aspects of CJD, but also from the patients that you've all studied at the Memory and Aging Center at UCSF. We will have to move to Eric because he's, I guess he's going to be in another meeting in about 15 minutes. I don't know if you can put the presentation, Eric.
Yes, great, thank you.
Michael, if you are still available in the following 15 or 20 minutes, I can give you 1 or 2 questions, if that's okay with you.
Sure.
Thank you so much. And so we will move to Eric Minikel, who is a senior group leader at the Broad Institute of MIT and Harvard, and he's been working in the past years trying to develop a new treatment for prion diseases. So we'll listen to Eric with his presentation, Novel Therapeutic Approaches for Prion Diseases.
Thank you so much, Victor. Really happy to be able to talk to everyone. Thanks so much for having me. My goal today is to give a little bit of a primer on how we're developing drugs in prion disease with an example from a drug program that we're developing right now. And first, brief introduction. I'm both a researcher and, personally affected. My wife, Sonia, is a mutation carrier. Her mom died of genetic prion disease. And 12 years ago, we found out that Sonia inherited that mutation, and we changed our careers to become scientists and dedicate our lives to a cure. So we now run a lab at the Broad Institute of MIT and Harvard, where we have 14 people dedicated full on to developing new drugs, new treatments for this disease, with the goal both to treat symptomatic disease and prevent disease in people who are not yet sick. So we think that the best evidence is for lowering the amount of normal PRP in the brain as the therapeutic strategy. So if we lower that protein, then there's less of it to misfold. Maybe if we do it early enough, it will never misfold. And we can do that by aiming anywhere upstream. We don't care about which, this isn't about allele specific for which mutation. We just want to lower all PRP because most patients only have normal PRP. It just misfolds, but there's no mutation. And there's a variety of approaches we can take. So the first project we worked on was with Ionis Pharmaceuticals. They developed an ASO for prion disease that I'll talk about very briefly. But today I want to introduce a few other ideas of how we can tackle prion disease. And each of these are different modalities. So if you think about pills in your medicine cabinet, those are generally small molecules. They're probably less than 500 molecular weight. You take them every day. They wash out very quickly. You have to keep taking them. There's now a drug in development called a molecular gate by a company called Gate Bio, where they are trying to actually do that. It's still in preclinical stages. It's not in clinical trials, but I think that's really exciting. The ASO that we've worked on is an oligonucleotide where it has to be injected into the CSF through a lumbar puncture, an intrathecal injection. And the other program I'll tell you about today, which is an siRNA, is also an oligonucleotide. Then we also have gene therapies. So these are really large protein complexes designed to edit the DNA, which makes it a one-time treatment for life, but it requires a vector to get it into the brain. And so there's a lot of technology development there to both have the right mechanism to specifically edit the DNA, but also to be able to deliver it to all the neurons, the 100 billion neurons in the adult human brain. So these are the data from the ASO collaboration. We've worked with Ionis for a decade now, and you can tell a sad story or a happy story about this plot. The happy story is when we treat the animals with ASO, they live 3 times as long. And the sad story is they all eventually die of prion disease. So 50% lowering of PRP helps, but it's not enough to cure the disease or permanently prevent it. So we need to go to deeper levels of knockdown. Ionis is now in the clinic. So there's 16 sites worldwide that are doing a first-in-human trial of ION 717, an ASO to lower prion protein. And I think any outcome is possible. I have hope. I think this is really the right mechanism. On the other hand, there's so many reasons why this trial could fail, even though the mechanism is correct, right? It could be that the dose is too high and the drug is too toxic. It could be the dose is too low and it doesn't do anything. It could be they didn't get patients until it was too late. There's just, it could be they didn't design the trial right to see the effect they wanted to. There's an incredible number of risks in any new clinical trial, and we have to be prepared for any outcome. So rather than sitting around hoping for this to give a good result, we need to be working really hard every day to bring additional drugs into clinical development in the hopes that we get one or a few drugs that actually work. One thing we've learned from the ASO trial is that our patients really show up. Prion disease is a desperate disease. Everyone wants to try something to help their relative who's suffering. And the trial enrolled much more quickly than Ionis expected, and they had to pause enrollment in the trial. So that was bad news for people trying to enroll at that moment. but it's really good news when we think about needing to develop a second and a third and a fourth drug. This proves to me that we can find the patients to enroll all of those trials. Of course, we shouldn't be complacent about where our trial sites are either. Ionis chose sites in, the US, Israel, Japan, Europe, Australia, Canada. I think we should have a trial site in Mexico as well. I was just in China last week talking to people there about how we can get trial sites in China. I think this needs to be a really good global effort to find all the patients and enroll as many people as possible in clinical trials. But I want to talk today about how we get from the idea for a drug, like I want to lower PRP, to having a clinical trial and ultimately figuring out if a drug works. So we use the term preclinical to refer to all the steps that happen before you dose the first person. And we could talk about first picking what we want the drug to do, figuring out does it do that, finding the exact drug molecule, and then getting permission to dose it into people. So when we think about selecting a target for a drug or a mechanism for a drug, we have to remember that the vast majority of drugs fail. Many of them fail because they don't do the right thing that's relevant to the disease biology. And in a common disease like Alzheimer's, where there's billions and billions of dollars being spent on drug development, maybe we can afford to take some shots that have very low probability of success. But I think in prion disease, a rarer disease, we need to be judicious, and we should focus on the mechanisms that are most likely to work. And for us, I think that's lowering PRP. That is the thing for which we have the most evidence. So all different lines of evidence from different areas of scientific inquiry all lead us to PRP as the cause of prion disease. We have a lot of evidence that if we lower it, that should be helpful. If you hear about other drug development efforts, ask how many of these boxes does that target check? Usually it's not many of them. All of the evidence really points us to PRP. So then let's talk about proof of concept. So how can we prove that lowering PRP with our drug is going to help? And for this, I want to introduce a case study. Sonia and I are working on developing a divalent siRNA to lower PRP. So divalent siRNA is a new type of oligonucleotide that was first introduced in 2019. It's never been tested in humans before, but these mouse and monkey data were published in 2019, and it looks really promising. In some ways, this siRNA is very similar to an ASO, so it also has to be administered into CSF intrathecally. It also cleaves the RNA, causing less protein to be produced. However, it seems to have really good potency. It can lower the protein very low. It lasts for a long time, and it distributes broadly throughout the brain. So we think there's enough reasons to be excited about this that it's worth having a second shot on goal in addition to the ASO. So we started by screening in cells, right? We grow cells in a dish and we test the compound to see if we can find an siRNA that will lower the mouse version of the gene. And sure enough, in cells, we could find an oligo that would lower PRP by about 90% in cells. But of course, things never look as good in an animal as they do in cells. So in an animal, we could lower it 50%. 50% is not better than what an ASO can do, but it allows us to ask the question, does lowering PRP by this mechanism work in our disease? So we then take that into prion-infected mice. We can see that when we infect the mice and don't treat, they die within 5 or 6 months. And when we do treat, even a single dose of drug, they live 2/3 longer. And if we dose chronically, they can live more than twice as long, almost 3 times as long. So lowering PRP by a different mechanism besides an ASO also affects disease course in a similar way. So then we need to find a more potent molecule that can be our human drug candidate. So again, we start by screening a lot of things in cells. The best ones we take into a mouse. We screen them in the mouse brain. Then we do a ton of different studies. We do dose response. We test different chemical modifications on the oligo to try to make sure that we have the best possible drug candidate. And eventually we say, that's enough. We've got a thing that looks really good. We want to take it forward. So we spent 3 years just finding what we thought was the best molecule. We started this project 2020, and it was by last year, 2023, that we had the lead molecule. So then we get to what we call IND enabling studies. So IND stands for Investigational New Drug. And investigational new drug, or IND application, is what FDA in the US makes us submit to ask for permission to dose a human. And the standards for what you can do in a human clinical trial are much, much higher than doing an animal study, right? So you need a lot more material to be manufactured, and you need it to be manufactured GMP, good manufacturing practices, which means very high standards of purity and identity and so on. This is a new type of chemical compound. Not many places can manufacture it. We ended up working with Hongene. They did a fantastic job. But manufacturing the white powder is just the first step. Then you have to formulate that into a sterile injectable liquid. You have to go do toxicology studies in 2 different animal species to see how high can you dose it before you get toxicity. You have to look at, does it modify DNA and have a potential to cause cancer or reproductive harm? Does it interact with other drugs that patients might be taking? All of this stuff costs more than $2 million and counting. And we did a shoestring budget. So this work was funded by NIH, and it's not done yet. And so, you know, it's still possible that this could fail. We may never dose a person if we get a bad toxicology result. But most pharmaceutical companies would spend easily $10 million on this. Developing a new drug to get to humans is really, really difficult. We had to do a lot of trade-offs in terms of how can we move this faster. It means that the first trial will be only single dose in symptomatic patients. We thought that was better than just keeping people waiting. We went to FDA, we asked really tough questions. We said, can we only do rats and not have to do a second species? They said, no, we do have to do 2 species, but they were flexible on some other things. They allowed us to use less of our drug on quality control testing. I'll skip over some of this. We have manufactured the drug. It's in the sterile injectable vials, but we don't yet have permission to dose a person. We're still waiting for toxicology animals to come down before we know if this is safe to proceed to a clinical trial. So right now, our goal is by the end of this year, end of 2024, to be writing the 1,500-page document that we submit to the government to ask for permission to start dosing humans. So it's a ton of work. Then we have to figure out how to do the clinical trial. We're applying for funding for that right now. It's been a real learning curve understanding how to do all of this, and it helps us appreciate why not that many things get to clinical trials, and when they do, it takes many years. But we think that this is a fight worth fighting because we can't just sit around and hope for the ASO to work. We need more drug candidates, and we need better drug candidates. So I have 1 minute for questions, and then I need to run to my next meeting. I'm so sorry.
Thank you so much, Eric, for this great presentation. We've already heard some news from Nina, and who's also working on a drug.
Yeah.
So how stimulating has this been for you? That you were not in this field and now move into a new one and try to develop a drug, and you're almost there.
Yeah, I mean, almost there is being generous. It could still fail for many reasons. And getting there just means being able to dose the first patient. From when we dose the first patient, it's still a very long road to know if the drug is safe or effective. And I always tell people to remember that all drugs do some harm, right? All drugs are somewhat toxic, have some side effects, and most drugs never do any good for anyone. And even a lot of drugs that are approved are not actually that effective and not actually that safe. I'm really hopeful in the aggregate about, I think PRP lowering is the right thing. And I think between what Nina presented you earlier, the ASO, the divalent siRNA, other things that are in the pipeline, I think we've got a real chance here to make a difference in the next 5 or 10 years. But I can't put too much hope in any one drug development program because there's just too many ways to fail.
Thanks, Eric. I don't want to hold you anymore. I know you have another meeting, but I know that we can send you or email you the questions from the audience, and
yes.
I know you will be very happy to answer them. Thank
okay.
you.
Yeah. Thank you so much.
Eric,
take care.
Thank you very much, Eric. Thank you.
Before going with Debbie, who is the president of the CJD Foundation in the U.S., I would just like to thank Michael Geschwind, and I guess you already answered one question about what are the how would you compare CJD with Alzheimer's, and I will also I also have one question, which is have you found clinical difference between early onset sporadic CJD and the common late onset sporadic CJD?
So can you repeat the question? Have I found differences between the early onset and late onset?
Yeah, is there a clinical difference between early onset sporadic CJD or and late onset sporadic CJD?
Well, yeah, I think other people have really done that work. So there have been a lot of people who've, they looking at large national data sets, and they subtype. So most, I gave you the average age of most CJD, but there are at least six, depending how there are ways of subtyping the prion disease based not only an individual's codon 129 polymorphism, but also the molecular weight when you extract the prion protein or the prion, the misfolded proteins from the brain at autopsy, and you put them on a gel and you digest it with proteases. The part that's resistant to protease digestion, you run it on a gel. If the lower band runs at 21 kilodaltons, they call that type 1. If it runs at 19 kilodaltons, it runs at, so it's a little smaller, they call that type 2. And you can get at least 6 plus different subtypes of that, like MM1, MM2. And it turns out that the VV1 is probably the least common subtype. And those patients, their median mean age of onset is about 20 years younger than most of the other cases. So so those tend to be the earlier onset ones. But then there are others that are earlier onset. We just had an MM2 thalamic subtype, which some people also call sporadic fatal insomnia, but it's just the MM2 thalamic subtype. We just had a 33-year-old with that onset, so very young onset. So yes, there are differences that can, and sometimes the MRI can show, the MRI pattern can, for some of those subtypes, it can help you identify which subtype it is while the patient's still alive. It's not perfect, and some subtypes, the MRI pattern doesn't really help. But it's a way for us to kind of prognosticate. It might be a way for us to think about stratifying patients in treatment trials, because we can't get the prion typing until we have brain tissue, which would either mean brain biopsy, and you'd probably have to do multiple brain biopsies, or autopsy. And so all we can get now is through just a simple genetic test by blood or buccal swab. We can determine the genetics, we can determine if they're genetic mutation carrier or not, and we can determine their codon 129 polymorphism. And then with other information like the MRI pattern, then we have a better idea of how to classify them. Are they going to be a fast progressor or slow? Are they more likely to present with uncoordination symptoms or more cognitive symptoms? But that's work that other people have done.
Yeah, thank you so much, Michael. We need to move on with the rest of the speakers, and I guess we will now move with Debbie Jobs, who is the current president of the CJD Foundation in the United States, and who's also a member of the CJD International Support Alliance. But Debbie, as the president of the CJD Foundation, works in a conjoint form. I don't know if that's the correct word, with Dr. Brian Appleby, who is the president of the CJD Surveillance Center. So I guess David's gonna speak first, and then it's gonna be Brian Appleby, and I guess we will learn a lot on how the foundation and the surveillance center can work together to improve diagnostics, therapeutics, and so on in the CJD field. So Debbie, please, we will listen to you, and we're very grateful to have you here today in this Mexican Symposium on Prion Diseases.
Well, thank you so much for inviting us. Glad to share a little about our mission and our programs. Which now are not advancing. Okay, there we go. So just a little about our key programs and how we work together with the National Prion Center. Really everything we do starts with the family and starts with helpline contacts. So we hear from families by, of course, email, phone, and through our website. And we get about 1,000 contacts each year, and we also speak with medical professionals, media, and the general public. So when we hear from a family, often they have just heard of the disease, just been diagnosed, or the case is suspected. So they really need a quick primer on what do we do about caregiving, what are these tests all about, and more. So one of the things we do is we educate them about the various studies and programs at the National Prion Center, which you'll hear more about from Dr. Appleby. One of the things we have to unfortunately talk to them about very quickly is the autopsy program because of the benefit to the families and science of that program. We also refer to various studies, including an MRI study and a great study Dr. Appleby does, which he calls the Teleneurology Assessment Program, where he meets with families remotely. And of course, we also make referrals to some of the studies you may have heard about earlier, biomarker studies and Dr. Geschwin's study, and fortunately to the clinical trial. We also invite family members to peer support groups and answer their other questions as well. So we offer close to 50 support group sessions a year. Virtually every week of the year, there's a session, either for current caregivers, for people who are bereaved, for those in different time zones. We have a couple of programs for genetic families, one for people at risk and one for caregivers. Almost everything we offer, from recorded lectures to fact sheets and more, is on our website. But a lot of families find it helpful to have this sort of bedside guide, so we do also send them a printed version. We've been offering families also these companion cards so that when they're in public with their loved one, with the patient, they can easily explain symptoms. So if they're in a restaurant or on an airplane or what have you, this is a helpful tool. And families are very often referred to hospice very quickly, but they also have a lot of questions about end of life, and we often share this series with them as well. This was written by a hospice nurse. I mentioned our website, and we recently upgraded it. It's a great source for information about all of our programs and announcements and things like the IONIS study. But it also contains quite a lot of suggestions on caregiving and practical things for the caregiver. You may have heard about our annual family conference, which traditionally has been in Washington, D.C., and is next year going to be in the Chicago area. One of the greatest things, having been a family member myself before working at the foundation, all of us love the moving roundtables. That's a chance for families to sit in a small group with the experts who are speaking at conference and get their questions answered. So a lot of my aha moments as a family member have been at those moving roundtable sessions. We also offer support groups for both sporadic and genetic families. We have a bereavement session. There's a keynote, the basics of prion disease, reports from the CDC and from the Prion Center, and more. And we have updates from those who've received research grants from the foundation. This year at our conference, we had a group of speakers who have actually published their story. One is a reporter at USA Today, one is a poet. These 2 books were written by family members as well. So Dave Langen captured the story of caring for his wife, and Don Friedman of caring for his sister. And of course, we had a talk from Amanda Kalinsky, who is featured in the Mercies in Disguise story about her family and her journey. And she is a family member who used IVF with PGD to avoid passing the gene on to her 3 children. She's been a great resource for families. We are happy to say that this year we awarded 6 research grants and 2 fellowships, and you can read all about the recipients on our website. So to date, we have awarded close to $5 million, and over the last 5 years, it's been over $500,000 per year that's been awarded through this program. We have a fellowship application process in progress, and it's still open through tomorrow if someone happens to be listening. That's the link to get the application, and those will be offered again next year. And our research grant application process will resume in spring of 2025, awarded the following year. We'd like to arrange and offer medical education with our volunteer speakers as many places as we can each year. And this year, my colleague and I visited the American Academy of Neurology conference, where we could interact with a lot more neurologists and spread the word about both our programs for families and the work of the Prion Center. Advocacy is a big activity for us, and it's yielding some results. The U.S. Congress, there are a lot of states, a lot of members. We have a lot of family members who come and visit with their representatives. And they share what the impact of the disease is on their family and the need for funding. And that's both surveillance funding and research funding. So we hosted an advocacy day on Capitol Hill last March, and we'll do that again next March. We also submit all the forms and testimony that's needed. And in the last 2 years, we've also done a resolution. So David Valadao, who's a congressman from California, spoke on the floor of the House about CJD Awareness Day, which is November 12th. Here are some of the families at Advocacy Day last year, and we look forward to welcoming them back next year to DC in March. So on CJD Awareness Day, which is November 12th, we will promote the resolution that Congressman Valadao made, along with others, as well as other social media and awareness raising. So it's a great way to rally families, give them some tools to explain in their community what the disease is about and the impact on them. Another thing that came out of our advocacy efforts is the scientific conference that we're hosting next week, also during CJD Awareness Day, just to have some dialogue about the overlap between the mechanisms in prion disease and the mechanisms in Alzheimer's and related dementias. And at NIH, the National Institutes of Health in the US, they view them as different things, and we're really trying to broaden that definition to incorporate prion disease. So we're going to have some very recognizable keynote speakers, as well as about a dozen panels and poster sessions on the topic. There's also a live stream, so you're very welcome to join. On our website, you can register for the live stream. All of this activity is funded almost entirely through donations from families and their communities. And one of the great ways of raising those funds is through our Strides for CJD program, which happens every October. So this year we hosted Strides in 21 different locations nationwide and had a great response and turnout. But it's really through those families that we're able to do all of those activities I described. Next year's family conference is in Rosemont, Illinois, right by O'Hare Airport, July 11th through 13th, and we would welcome you to attend. So thank you very much. Hopefully this gave you a little bit of a slice of what we do and how we work with Dr. Appleby's Center.
Thank you so much, Debbie. It is indeed a lot of work that you all do at the CAD Foundation. You've given a lot of grants, you've helped a lot of people, and you work in a very, very good way with the CAD Surveillance Center. I must say that the only reason that I won't be in DC next week is because I'm also hosting a meeting at my university. When I saw that we had the same dates, I was like, oh my God. Because I guess it will be very, very helpful to start focusing into both Alzheimer's and prions and to see how we can work together, um, um, trying to find answers for the pathology or physiopathology of these diseases. I guess it's a very, very good idea.
Yes, we're hoping it'll further collaborations and dialogue and, you know, open the door to more funding for prion disease research for sure. But it'll be recorded and we're also hoping to publish some of the outcomes so you can catch up then.
We will definitely will. Thank you so much. I guess we'll have to move to Brian, who's also about to start a meeting. And so Brian, Dr. Appleby, who's the head of the CJD Surveillance Center. He's a physician, he's a psychiatrist, and he's been working with CJD patients for a long time, and who in the past years he was the chair of the head of the National Prion Surveillance Center. So Brian, thank you again for being with us, and we're ready to listen to your presentation.
Great, thank you, Victor. Can you give me a thumbs up if you can see the slides?
Yes.
Awesome, all right. Well, thank you very much for the invitation. I really appreciate it. I was told there'd be multiple people in the audience, family members, professionals. So I tried to include a little bit for everyone, but you know, we're always going to focus on the families first. So I apologize to some of our colleagues if this is more family centric than what they expected. So a little summary of what we'll review today. We'll talk about how prion disease surveillance is conducted in the US, because it is different depending on what country you're talking about. I'll provide a little overview of the Prion Center in the US and then present some basic findings. So interestingly, tomorrow, all the surveillance centers around the world are going to be meeting. And so this is kind of interesting that we'd have this talk too. And I thought it was just interesting to kind of review how the US does its surveillance. So there's multiple agencies involved. The Centers for Disease Control and Prevention really guide everything. They put out the final numbers. They fund the National Prion Disease Pathology Surveillance Center, and also they have a grant for the CJD Foundation as well for education. And then they also help fund some people in key state local health departments. And there's a variety of different activities that go about doing prion disease surveillance. The CDC collects death certificates, and then they take those certificates and cross-check them with our prion center data. Sometimes people are correctly diagnosed as having prion disease, sometimes not. And then sometimes it's something completely different on the death certificate. So we're able to add those cases based off of tests that were done at the prion center. The US has several different concerns when it comes to prion disease, especially from a public health standpoint. And one thing that I find interesting when I travel to different countries is different countries have kind of their own theme of why prion disease surveillance started in their country and what their initial concerns or current concerns were. So for the US, we've been somewhat fortunate in that we haven't of course, had the epidemic of mad cow disease that the UK had, but we have definitely come across acquired prion disease. So we've come across 42 cases that are medically transmitted, what we call iatrogenic CJD. Most of these were due to human growth hormone that was derived from cadavers. 4 came from dermatografts that also were derived from cadavers, and then we have 1 due to a corneal transplant. Variant CJD, which is the kind of CJD in a human due to eating meat contaminated by bovine spongiform encephalopathy or mad cow disease. In a human, we call that variant CJD. And fortunately, in the US, we've only had 4 cases, all of which were thought to have been acquired outside the US. The last case was detected in 2014. Unfortunately for the US, we have an epidemic of an animal prion disease called chronic wasting disease. This is a disease of prion disease in the cervid species, which includes deer, elk, moose, and caribou. And it's very easily transmissible amongst its own species. The prions are excreted in their saliva, their urine, their feces, and they contaminate the environment for a long time. Of course, these are also free-ranging animals, which brings a lot of difficulty in trying to contain such an epidemic. And this is really kind of the primary purpose of the Prion Center at this point in time, is trying to determine whether or not chronic wasting disease can transmit to humans, either through human consumption of venison or through a secondary animal like a cow. To date, we've had 35 states affected by chronic wasting disease. That is rising every year, of course. And I'm happy to report that to date, we have no evidence that chronic wasting disease has transmitted to humans. But as you've likely heard in prior talks, the incubation period for these illnesses can be quite long, usually measured in years to decades. So it may be that it's already occurred and we don't know about it, or of course it could happen in the future when we have different strains. So I'm going to provide a brief overview of what we do at the Prion Center. We were initially funded by CDC in 1997 at a response to the bovine spongiform encephalopathy epidemic and the identification of variant CJD in people. We reside at Case Western Reserve University in Cleveland, Ohio, and it was started by Pierluigi Gambetti. We conduct neuropathologic surveillance activities for the CDC because the only way to have a definitive diagnosis of the disease is through autopsy confirmation. That also is the only way to determine the etiology and the type of prion disease. So for example, is it sporadic, genetic, or acquired? And if it is sporadic, as Michael was talking about, what type of sporadic prion disease is that? So it allows for neuropathologic and also biochemical surveillance for novel forms of the disease. But as you'll see later, we also run a clinical lab on patients that are still living with suspected prion disease, and that allows us also to capture cases. As you could guess, an important focus is on preventable and potentially novel forms of the disease, such as those illnesses that I reviewed before, variant and iatrogenic CJD, and of course, novel acquired prion disease, specifically the threat of chronic wasting disease to humans. There are a variety of different aspects of the Prion Center. At the core of it is neuropathologic surveillance, the slide that I just reviewed, where we coordinate autopsies from around the country of cases of suspected prion disease. And then we also do neuropathologic characterization, so we know whether or not we've seen this type of prion disease before or not, or is this a novel prion disease that we need to be concerned about. We also run a clinical diagnostic lab where we can run clinical diagnostic tests on spinal fluid from patients. We do 2 markers of neuronal injury, 1433 and total tau protein, and then we do a very specific disease that detects the abnormal form of the prion protein called RT-QuIC, which stands for real-time quaking-induced conversion. That is extremely specific. And then we do genetic testing either on brain tissue or on blood. Most of our research in-house is characterization of cases for the surveillance component, but we also help with improving diagnostics, including spinal fluid testing as well as brain MRI. And as you'll see later, we have a very large repository of specimens that is important not just for our research, but for international research. As you heard from Debbie, we do a fair amount of outreach and education through presentations, publications, our website, and of course, through their help. And we provide consultations to physicians, state health departments. We have a free brain MRI consultation program, and then the TAP CJD program, which is that telemedicine longitudinal study that Debbie talked about. And then we also see it as a mission of ours to help guide and inform clinical studies and to help provide as quick access and availability to trials as possible. And of course, we're participating in these clinical trials, such as the IONIS study. I can't emphasize enough how important our collaboration with the CJD Foundation is for our purposes. It ensures first and foremost that families get the appropriate support. Our field is very medicine and researcher heavy, which is good, but it also can sometimes leave families in the lurch. So whenever we refer our patients and families to the foundation, we know that they're getting appropriate support to their level of understanding. They definitely help us with autopsy referrals, referrals to the teleneurology assessment program, as well as brain MRI referrals. They're very active in those referral processes. And they have a variety of educational events that we also help with, such as grand rounds for neurologists, internists, and that's from around the country, and then outreach events. So this is a picture of what Debbie was saying when we were at the American Academy of Neurology, and we were able to have a table right next to them, which was kind of nice because you have the family perspective and then you have the laboratory perspective. And of course, they have excellent support and educational groups for these families, and they've really gone out of their way to help develop educational content that are understandable for lay individuals, and we also help them with that. And then, of course, their annual family conference. Okay. So I'm going to show a couple videos. Again, this is mainly for families, but I think it helps emphasize the importance of what we do. The first is on our biorepository. This is we have over 8,000 brains that we've collected over the years. About 70% are prion disease, 30% are something else. And this is our new repository in which they're housed. And Victor, if you just give me a thumbs up if you can hear.
The National Prion Disease Pathology Surveillance Center proudly operates a 1,485 square foot repository that is located within the Cleveland Center for Membrane and Structural Biology. The repository continues to grow to meet the needs of the Prion Center and is currently equipped with 33 ultra-cold freezers that are storing almost 8,000 brains. This space allows the Prion Center the capabilities to support research efforts globally. Requests are received, processed, and selected for collaborating scientists. Staff maintain all freezers, and monitoring is achieved remotely 24 hours a day, 365 days a year. The system in place sends notifications to designated staff electronically. As a part of our inventory initiative, we are able to select brains for a variety of different criteria utilizing a barcoding system within our research management software system known as LabMatrix.
So as you can imagine, that biorepository is something that we're very proud of, as it really is a legacy for our families. And I think the families feel the same way, that their loved ones are able to continue to contribute to research of this illness. Next, I'm going to show you a video about some of the detailed workings of the Prion Center. I talked in general terms what we do, and now Gabby's going to take you on a tour in the lab.
The National Prion Disease Pathology Surveillance Center is the only center of its kind in the United States. The center coordinates autopsies and neuropathologic examinations of suspected prion disease cases from across the country. Local autopsies are performed on-site, and other cases are performed elsewhere through a network of providers that the center coordinates. In the field of prion disease, Western blotting is an important technique used to identify positive cases and their subtypes, as well as establishing the type of abnormal protease-resistant form of the prion protein, also known as scrapie prion protein. This procedure involves running proteins from patient samples and marking them with specific antibodies, allowing the lab to determine whether the case is positive and to identify the subtype. The histology lab sections, processes, and embeds tissue for histological and pathological analysis. They primarily utilize immunohistochemistry, allowing us to visualize the presence of prion protein within samples. Additionally, they use the H&E stain, known as the gold standard, to provide a comprehensive view of cellular and tissue structures for routine pathological diagnosis. On fixed brain tissue obtained either at autopsy or biopsy, the histology lab excludes or confirms, then characterizes the prion disease by microscopic examination following ordinary histological procedures and analysis of the prion protein, as well as a pattern of tissue distribution. In the CSF lab, prion marker testing is performed through a prion panel, 1433, tau, and RT-QuIC. The 1433 protein is a marker for some prion diseases, such as CJD, when a number of other neurodegenerative conditions are excluded. The CSF team measures total tau in CSF, which is markedly elevated with prion disease. RT-QuIC, real-time quaking-induced conversion, is a high specificity test that is used to detect abnormal prion protein. The molecular genetic lab searches for the presence of a mutation in the prion protein gene and determines the polymorphism at codon 129 and at other codons. This is tested through DNA extracted from blood, brain, or any other tissues.
Thank you to Gabby for putting that together. So I'm just going to end with some basic info on number of cases in the US. So this is a graph of all of the different brains that we've received over the years. The top yellow line is the total number of brains that we've received over time. The one below that, the orange line, is the number of prion positive cases. So what you'll see is there's about 30% of brains that we receive that wind up not being prion disease. And there's a lot of variety actually of what they wind up being. Most are other neurodegenerative illnesses like Alzheimer's disease, but some are unfortunately treatable illnesses. The line below that, the red line, is the number of sporadic cases that we have per year. And in our country, that makes up about 90% of our cases, followed by 10% of genetic forms of prion disease, the pink line. And then you will see that the acquired forms, the medically transmitted and variant CJD, are fortunately few and far between. One of the things that we can do with RT-QuIC, because it's so specific for prion disease, is we can count it as a positive case. And we do that. And as you can probably imagine, a lot of those people go to autopsy, but there are some people that either never have spinal fluid tested through us, or their CSF is negative, which it will be in at least 10% of cases. So what we can do is you'll see that the number of RT-QuIC positive cases every year is about 500. And then if we add that to the number of people that had brain autopsies that didn't have spinal fluid results, it increases it by almost 100. So this top orange-red line is the number of prion disease cases that we have per year in the US, which is close to 600, which is about what we would expect. And that's remained fairly stable over the last 4 years. So in summary, I just want to end with saying that in Victor, the families have an extremely energetic and enthusiastic advocate. He's been in the field as long as I've been in the field, and he's always at meetings representing the families. So you should be very proud of him. And Victor, thanks again for inviting us.
Thank you so much. Brian, for this talk. I don't know if we have any questions from the audience or Francisco. No. Brian, besides having the brains at your biorepository, do you have any other tissues like lymphatic tissue or I don't know?
Yeah, we have a variety of different tissue types. Of course, we have over 8,000 brains. We probably have over 100,000 spinal fluid samples, a fair number of blood samples. And as you mentioned, we do also collect what we call lymphoreticular tissue. So these are different organs that deal with the blood supply in the body, like the spleen, appendix, and lymph nodes. And we do that on cases that may have been exposed to chronic wasting disease through venison consumption or hunting, because like we know with variant CJD, that also resides in those tissues. So a small amount of those, but we do have some of those.
Yeah, and are those tissues randomly selected, or you go or are you guided by the clinical of the patient?
For the lymph tissues?
Yes, yes.
Yeah, so the lymph tissues are obtained from patients, of course, with the family's permission, if they hunted or consumed venison in a state that's known to be affected by chronic wasting disease.
And one last question. I know you gave all of these numbers from the surveillance, and we know that you don't only process samples from the U.S. How many samples? I mean, what will be the percentage of the samples that you get from all over the world?
Oh, I wish I'd looked that up. That's a great question. For spinal fluid, I would say it's about 10%. We get a lot from Brazil, Taiwan, and Hong Kong. Our brain specimens, though, are almost universally from the US. We do collaborate with other international surveillance labs to do like validation testing. But aside from that, we would send them to their appropriate surveillance program in their country.
Yeah, and that's one of the issues in this continent in America is that it's basically only the U.S. and Canada who have a proper surveillance center. So most or all of the samples that are from the patients that are studied in Latin America, they basically go to the surveillance center in the U.S. So we are also grateful for having you helping us trying to diagnose a patient in Latin America. We're working on establishing a lab. You know that we've been trying to do that for a lot of years. We guess we will luckily be there in 1 or 2 years. But so far, I will say that most or all of the samples that have been studied in Latin America have been studied in the surveillance center. So that's another weight of gratitude that we need to give to you. That you don't only focus to the U.S. but also to all of the samples in Latin America and in the world. Thank you so much, Dr. Brian Appleby, for this presentation, for helping us getting some of the or most of the diagnosis in our countries. I don't know if we have any more questions, or I know you have to go into a meeting, Dr. Appleby. I don't want to hold you any longer, but if there's any question, we can email it to you, and I know you will be very glad to answer that.
Yep, I'd be happy to just send me an email.
Thank you so much.
Thank you. Enjoy the rest of your meeting.
Thanks so much.
Thank you.
Okay, so we're almost done. We are very happy and again grateful for having all of these international speakers in our symposium. I know Michael Geschman is still with us. Thank you, Michael. I guess you already answered some of the questions that were left in the chat. I saw another one. If you still have time, Michael, that says, how could we treat? Because when you mentioned this patient about the posterior cortical symptoms, So they were asking how is there a way of treating those specific cases or treating posterior cortical atrophy patients who may or may not be related to CJD?
Well, so posterior cortical atrophy, or PCA, is a clinical description of disease that tends to affect the posterior parts of the brain. And that can be due to many diseases. Most commonly is Alzheimer's disease, often an earlier onset form of Alzheimer's. But we can also see that in dementia with Lewy bodies, which is a disease similar to Parkinson's disease. It's caused by the same protein, alpha-synuclein, that we see in Parkinson's disease and multiple system atrophy, all alpha-synuclein. And probably the third disease, well, actually, the third disease would be cortical basal degeneration, which is usually due to a protein called tau that misfolds. And then fourth would be due to CJD. So when a patient presents with a posterior cortical atrophy syndrome, they often have a lot of problems with visual processing, with visual spatial skills. Our first job is to figure out what's the cause. Is it underlying Alzheimer's, cortical basal degeneration, Lewy body, or CJD? Because each of these is probably going to have their own treatment. We're not going to take one of Eric Minical's treatments for prion disease, where we're trying to lower the prion protein to treat Alzheimer's or alpha-synuclein. And the mechanisms that Dr. Minical spoke about, actually for prion disease, the same methods are being tested in laboratories around the world for the other diseases, the other neurodegenerative diseases. So trying to reduce the level of Huntington's disease or alpha-synuclein. I don't know if I answered that question.
Yes, yes. And I was I also had a similar question when you talk about this patient with the posterior cortical atrophy, because that's a symptom that we see a lot in our early onset Alzheimer's disease population. So considering this case that you mentioned, were there any other findings besides prions? Because there's been some cases that where they find beta amyloid pathology with prion pathology.
Can you ask the question again? I think I understood it, but I don't want to answer the wrong question.
Yes. In some cases where when they are treated either as Alzheimer's or prion, when they died and you get to have the autopsy, you find beta amyloid deposition and also prion deposition. So could this case that you showed us, or some of these cases where they showed that the posterior cortical atrophy, have a pathology of both beta amyloid from Alzheimer's and prion from CJD?
So we did a study, I shouldn't say we, one of my colleagues, Dr. Steve DeArmond, he's retired, but he looked at 256 of our autopsy-proven CJD cases. And what they found is that about 20% of those cases had amyloid pathology as well as CJD. So it doesn't mean that they met diagnostic criteria for Alzheimer's disease, but that they had a significant amount of Alzheimer's pathology, at least enough to be detected under the microscope and with immunohistochemistry. And it's more than we would have expected for that age group. So there might, and there is evidence that, for instance, that the amyloid protein receptor in the brain, that the prion protein seems to affect the bringing of amyloid into cells. So there probably are some interactions going on between these proteins. I think it's really an excellent point that there's probably, and then work by Simon Mead and John Collinges and others in their group in the UK, there's because of the problems with human growth hormone, what they did is they actually looked at patients who received growth hormone and they looked to see if they had more Alzheimer's pathology than expected. And they claimed that they did. It was published in a very big journal, but my feeling from that paper I wasn't as convinced as they were by the data. I didn't think that they did find some patients with Alzheimer's pathology that were younger than expected, but I don't really think they had enough to be considered Alzheimer's disease. That being said, there is some indication that these other proteins, amyloid, A beta amyloid or A beta 42, alpha synuclein, they might be transmissible from person to person, just like the prion protein is. If you take extracts from a brain or the hormones from a brain, you might be able to transmit the proteins. But it doesn't look like you can really transmit the disease like you can with PRP prions.
Okay.
Excuse me.
Okay. Thanks, Michael. I guess we will move on to our last presentation, which is from Francisco, who's the president of the Mexican Foundation. I already sent you, Michael, his presentation, so he translated it. So if there was still any of the speakers with us, so you could follow his speech. So, and I will say this in Spanish. El arquitecto Francisco, desde hace algunos años, ha estado trabajando de manera intensa por establecer una fundación para priones. A final de cuentas, por cuestiones gubernamentales, la fundación se llama Fundación Enfermedades Neurodegenerativas México, en donde ha estado apoyando los esfuerzos para el abordaje clínico y también en investigación en personas con Alzheimer y con enfermedades por priones. Francisco ha estado también trabajando para establecer el primer laboratorio de diagnóstico para enfermedades por priones, no solo en México, sino en Latinoamérica, con la esperanza de que pronto podamos ofrecer lo que ya ofrece el Centro de Referencia Epidemiológica para Enfermedades por Priones de Estados Unidos, obviamente no con las grandes dimensiones que tiene hasta el momento con 8,000 cerebros, ¿no? Pero sí empezar poco a poco a ofrecer esta esperanza diagnóstica y eventualmente ligarnos a los ensayos clínicos para ofrecer una esperanza de tratamiento a estas familias en el país. Sin más, escuchamos al arquitecto Francisco Mota, su presentación sobre la Fundación FENMEX y con esto concluiremos nuestro simposio de hoy.
Víctor, muchas gracias. Pues trataré de ser breve y haré un pequeño resumen básicamente de lo que es la fundación y pues digo, y el panorama en México, básicamente. La Fundación de Enfermedades Neurodegenerativas México, que así se llama, también llamada FENMEX, es una fundación sin ánimo de lucro. Es una entidad cuyo fin no es la persecución de un beneficio económico, principalmente persigue una finalidad social, altruista, humanitaria y o comunitaria. Este tipo de instituciones por lo general se financian gracias a ayudas y donaciones derivadas de personas físicas, empresas e instituciones y organizaciones de todo tipo. En algunos casos, no en todos, también se recibe ayudas estatales, puntuales o regulares. A partir de las proyecciones nacional de población para el año 2050, 32.4 millones de habitantes serán personas mayores. En México, a partir de los 65 años, una mujer de cada 4 y un hombre de cada 5 se ven afectados por problemas de salud neurodegenerativos. En total, se estima que más de 1.5 millones de mexicanos sufren de alguno de estos padecimientos neurodegenerativos. El humano está evolucionando, cambiando sus hábitos y costumbres, por lo que su calidad de vida se está extendiendo, la longevidad. Pero no todo el cuerpo se adapta. La parte más importante de él, el cerebro, se deteriora y en consecuencia limita al ser humano en sus actividades. Enfermedades como el Alzheimer, Parkinson, Huntington, CJD, demencia senil y esclerosis lateral aminotrófica son las principales en México y en el mundo. Afectan al sistema nervioso y ocasionan pérdidas de memoria, alteraciones de la conducta y funciones cognitivas. Actualmente la Organización Mundial de la Salud estima más de 30 millones de pacientes en el mundo con Alzheimer, duplicándose esta cifra cada 5 años. Durante la última década se ha producido un cambio hacia la atención en las intervenciones tempranas para maximizar la ventana terapéutica a través de medidas de prevención primaria que actúan sobre los factores de riesgo accionables o medidas de prevención secundaria para frenar la progresión de la enfermedad, según afirman científicos del área. Especialistas, instituciones farmacéuticas y fundaciones trabajan para poder ayudar a las personas que padecen estas enfermedades en busca de formas de prevención, tratamiento y, en su caso, cura. ¿Para qué pasa? Pero ¿qué pasa con los familiares de estos pacientes? Son ellos los que cargan con los gastos y cuidados en la mayoría de los casos. Familiares que, aparte de realizar estos trabajos, tienen la incertidumbre de portar genéticamente esta bomba de tiempo. FEDMEX no se olvida de ellos y, además de proveer apoyo a los pacientes de estas enfermedades, se enfoca en apoyar a los familiares. En la mayoría de los casos, los familiares son los que más sufren y es urgente desarrollar procesos, manuales, agendas y canales de ayuda. Para ello se requiere una cantidad de recursos que, además de que estén enfocados a lograr una sustentabilidad de la fundación que lo recibe y distribuye, para lograr esta sustentabilidad y además estar a la vanguardia de los últimos descubrimientos de este ramo. FEDMEX estableció los canales de comunicación con las más grandes fundaciones especializadas en este ramo. Esta unión hace que las investigaciones y resultados fluyan de una manera orgánica en beneficio de los pacientes y familiares. Aunado a este mecanismo de información, también se establecen las fechas de encuentro globales en forma de convenciones, donde la interactividad real y virtual tienen efecto, lo que permite tanto a estudiosos como a pacientes compartir sus avances y experiencias, como lo acabamos de ver hoy. Necesitamos generar la participación de todos los sectores de la población. Los años avanzan para todos sin excepción y es importante no olvidarlo porque después puede ser tarde para recordarlo. ¿Qué servicios ofrecemos que no existen en otras fundaciones? Podemos dividirlo en 5 grandes temas. Apoyo para las familiares y pacientes, así como el voluntariado que se vaya uniendo. 2, prevención con la capacitación y entendimiento a familiares, amigos, enfermos, empresas que deseen tener una cultura entre sus colaboradores de estas enfermedades. Dar a conocer a mayor detalle las consecuencias de cada una de las enfermedades neurodegenerativas nos dará una cultura distinta y si ya la conoce te enfrentas a estas sin miedo a todas ellas. 3, congresos nacionales e internacionales. Aquí tendremos la oportunidad de tener los grandes médicos en cuestiones de enfermedades neurodegenerativas con sus avances médicos y científicos. 4, investigaciones de los adelantos científicos y alternativos que se están desarrollando y o gestando en cualquier parte o rincón del mundo. Y el 5, establecer convenios con instituciones educativas de investigación y asistenciales. FEDMEX a la fecha ha atendido a 19 familias de diferentes partes de la República en estos 2 años de atención, tratando de apoyarlos con información general y en algunos casos particular. Todos han sido atendidos por el doctor Víctor Javier Sánchez González. Se requiere de un equipo mayor de profesionales para el apoyo, como son un grupo de médicos, psicólogos, psiquiatras, para guiar con mayor facilidad a las familias afectadas. Estamos agregando en breve a un mayor personal para ello. Agradezco mucho su participación en este segundo simposio. y esperamos poder seguir haciéndolo en esta labor tan importante y dar seguimiento a los avances científicos de esta enfermedad. Nos vemos en un año. Gracias, gracias a todos. Víctor, gracias.
Muchísimas gracias también a ti y pues bueno, ya clausuraste el evento, yo creo que no tengo nada más que decir.
Adelante entonces. Pues agradezco a todos su participación. Muy amables.
Muchas gracias, nos vemos el próximo año.
Hasta luego.